with this presentation, which is a general um, panorama of what we are looking today. The professor who will be presenting is Jorge Cortez. He is from the Central Investigation Center in the Biology School for Ocean Sciences and Limnology of the University of Costa Rica. He also is in different researches for marine investigation in different areas of Costa Rica. He has published several works of his investigation. His purpose is to create more consciousness, more awareness of the importance of oceans in Costa Rica. So you have the floor. Good morning, and thank you for the invitation. We will begin. These slides are in English, but the presentation will be in Spanish, so that what we will be speaking today is regarding the state of knowledge of the marine biodiversity of Costa Rica, and I will speak about Costa Rica's different ecosystems and its richness and marine biodiversity. As many of you all know, most part of our country is actually ocean. And this map, um, we can all recognize it perfectly. And it took almost 30 years for the government to define that this is the map of Costa Rica. And it makes us the biggest country in Central America. And the list goes on as to comparison. And a lot of that is because of the Coco Island, Isla del Coco. We not only have a large ocean area, but it's also very deep. 63% is below 2,000 meters of depth. 36% is below 33,000 meters. And then the deepest point is 5,250 meters, which is a lot more than the Chiripo, which is 3,820 meters. We have a lot of ecosystems. We have the Dulce Gulf, which is a basin. We have many different ecosystems with very fine sediments, sands, beaches, rocky intertidal mangroves in the Caribbean and in the Pacific, seagrasses, also in the Caribbean and the Pacific, and then coral reefs as well. And that's one of the reasons why we have so much biodiversity, because we have coast in both the largest oceans of the planet. We also have some rotoliths beds. They seem simple, but they are very complex. The Coco Island, it's open ocean. We also have open ocean, some of the largest animals that exist in the plankton. We see some seamounts of all sizes, from low monticles, from mountains of 1,500 meters with the same depth. Methane seeps. In the Pacific, methane is toxic, but the bacteria obtain that and use that as a source for feeding. And then we also have the abysmal plains, which are the really extensive mud floors. And it seems simple, but that's really full of life. And that's in front of Guanacaste. In 2009, with our colleague, we edited this book And what we did was invite different specialists as to what was recognized in the country, and we reported 6,778 marine species, more in the Pacific than in the Caribbean. And to the date, while we look back at everything that has been published till now and what is in progress, and some of the projects that we will mention here, I believe we are among around 7,300 species, maybe even more. A lot of new species. It's a very large list of new species as well. And this is just an example that Dr. Jeffrey Cibaja Cordero of worms from the Coco Island. These are just pictures of the heads of the worms, and this is a publication before 2012. And then so we gathered all the information that we had before that, and it was 6 families, 9 species. And by 2012, we were with 35 families with 106 species. And we had begun the project in 2007, 2009. We started a big project in the Coco Island. And for now, I consulted with Jeffrey, and I think that we're around 47 families and 163 species. And this is an example of what different studies make, studies by different groups in different areas. And it's such a large gap, and there's a project that somebody will speak with regarding in just a little bit more forward. And we have so many species now. These studies make a very big difference. And to conclude, I would also like to emphasize the importance of the marine protected areas. It's the last refuge for many species, and we need to make sure that several areas are created that have that protection and conservation. And every time we go someplace new, we're finding new species. So we're just beginning, really, to understand and to become aware of what we have in the country. And as a conclusion, Costa Rica has a huge marine richness in environments and species. We do need more studies. And not only studies, but also monitoring what is happening. Not only, oh, that species is there, but what else is there? How is that changing? We need to reduce anthropogenic impacts. We find trash in several meters of, of death, and that shouldn't be happening. We need more and larger MPAs and more protection. We need more protected marine areas, and that's quite a subject nowadays. They actually want to reduce the ones we have, and we actually need more. We need to take care of the ones that exist, better them, and create new ones. And therefore, we need more protection. I don't know if you all know that the Environment Ministry for Conservation was reduced by 40%. And while Incopesa, which is the exploitation ministry, was actually increased by 60%. So we're breaking down the focus and mission that this country has, which should be conservation, that be in land and also in the sea. So yes, that is it. Thank you. Thank you. And we will move ahead with Dr. Jeffrey Cibaja with the presentation called the Biomar Project, which is the project that Don Jorge was mentioning. Jeffrey has a license in biology from the Costa Rica University in water resources. He also has studies in Vigo, Spain. He works in 2006. Since in the University of Costa Rica, he's also a teacher, and he also works in CIVET since 2013, and from 2020 to 2024, he has been also director of the Biology magazine. Okay, well, thank you. Good morning. I came to speak about this project as an example of the different projects of biodiversity and how we have led it. The project is called Biomar, and this is a project we have with the project of conservation of Guanacaste and CIMAR, which executes it. And we also have an alliance with different other schools and museums. Next slide. This project came about due to a conversation between Don Jorge, Danielle Jansen, and Danielle is more focused into conservation of some land environments, and Don Jorge is more focused in water conservation. So they um, they gathered up and we determined a project to explore this marine area with some publications, some explorations, specific explorations, but not so much as an inventory. And this is something that we did in the area of Concheniquil, and that's where it began. I joined the project in 2016 to begin the investigation of those worms, and Don Jorge coordinates this, and now we do this together with along with Fran Joyce, which is the NGO in Guanacaste. So we have those three coordinators. Next slide. And this is developed in the conservation area in Guanacaste. We have this sector here that has a coast that has different levels of conservation. There's an area that has a more marine conservation, which is the blue one, that has more conservation, and then we have the marine landscape, which is a lot more area, and that stems up to Maria to the Culebra Island. And so we've covered most of the coast, and we will continue with more deep areas. And so we look for different organisms. These are some examples. Hydroides, marine sponges, poliquetos. They're called small Christmas trees. They stick on coral reefs. And so then we gather that and study the species. These are other species that we've found. We take a picture, we identify them, we present them like this. Everyone who is up here, who's sitting in the front, is involved, and they have their own team, and we move ahead with that exploration. And we have some mollusks, some marine slimes. We have some crustaceous, and there's a student, Carolina Salas. She will begin to study that. And I was working with the poliquetos, and there's a lot of groups. I can't really present them all here because of time, but the idea is to take a picture of them while they are there alive. So we are here, and we can see the idea of we can get an idea of the different participants of the community. Before this woman here, she was a housewife. She had her family. She cared for them. She had some fishermen family, but she wasn't really involved. And then we also have Hubert Ampie. He was a local fisherman. They go on ships, and they also dive to look for octopus. And so then we decided to create these actors, which are people on land that help and assist the investigators. And this is an example. These two people, they joined the group, and they have been a lot of help. Now they handle data, they extract DNA, sampling, and they're a great assistant. She is someone who also helps in logistics. And then Hilbert, he was a fisherman. He knew a lot about the sea, but maybe he didn't really know some of the weird species that we presented. So that also opened up their eyes and their knowledge to different types of fauna who who um, who didn't know this, and so then they also shared this knowledge with the community. And so sometimes there are some other people that come forward and that know them, and they maybe want to also be involved. If they maybe just see some strangers, maybe they're shy or they think it's something completely different, but when they see community members, then they come forward and then they also get involved. So they are great support. The taxonomers, parataxonomers, they also look for samples in the beach. Next, we also go on different ships in the Murcielago Island. We have some tools here that drag, that we've dropped that to 20 meters, and then we lift that, and we have a lot of other fauna that we see here. And then we see here different microhabitats from that are in the sand, that are in all types of environments, to look into all types of species and micro organisms that we can find. And then this man here, he takes the pictures of the live microorganisms and the mollusks, and some investigators take pictures of fishes, but most pictures are taken by Hilbert. He also learned the photography in a more professional manner, and he's a great support in that sense. We need to do it quickly because some microorganisms are weak and they can die very quickly within minutes or sometimes hours. So that needs to be done quickly, and that's in Murcielago Island. And then we also have an heteroscope where we look into some smaller organism. This is a student, Beatriz Orquidon. She's going ahead with her thesis with some different species and different subjects. And then some other, next year I'm sure we'll have other students that have a specific focus. And so then they also have the support of the community members. And sometimes it's late, sometimes it's nightfall, but we're still working, taking pictures. They have the laboratory that sometimes are in the fields. Sometimes we improvise a bit, or we just set it up in the beach, and then we just do the work there. Next slide. And then there we obtain the samples to obtain the DNA for code analysis and the collections. And then there we need to see what we're still missing for their genetic ID. So we obtain a fragment of that, and then we take that to the CBIT.
Mentioned this collaboration with the Guanacaste Conservation Area. We have the individuals such as Roger Blanco, Maria Marta Chavarria, working with us, and we to counsel with them to see what areas are of priority to explore, and also coordinating all the other areas of interest. So we work tightly with them. Sometimes when we're there, some other groups that are in some type of activity come close and ask about the project. So we explain the whole thing to them. We have Gustavo Induni over there in the picture, who's from SINAC too. We have Frank Joyce too, who's the other coordinator for the project uh, for the Dry Forest Conservation Organization, and has provided a lot of logistical support. Also, the Dry Forest Foundation is a very, very good financial support for this whole project. Here we have different individuals from the foundation, where we have Eric Balola, Monique, Hilbert as well, who support us in all of the logistical part of things. And this is a convention between the Ministry of Environment and the University of Costa Rica, in which the Ministry of Environment has a financial support specifically for the Guanacaste conservation area from this NGO to finance conservation activities. So this project is put into this three-way collaboration, plus the local community as well. So, for example, we have Yolanda here with some interested individuals who perhaps are interested in providing the area with donations for the project, and then and different individuals and staff from this organization. And as I mentioned, the samples are taken, they are placed in containers for DNA, and so far we have 720 species that have been genetically sequenced. No, Mr. Jorge mentioned 1,500 from the reports and what has been identified morphologically, but since we are matching with the DNA sequencing, then it's very likely for this number to be higher, because it doesn't mean that all of these 720 sequences are from those. They are spread between what was known and what is new. So, of course, we have more to sequence as well. Next slide. This is done in Canada along with WELP University, and that is assisting us with the sequencing, and of course they're paid a fee for this service. This is just a few of the people who are implicated, a large amount of students who help with assistance hours, teachers, and, educators. Almost everybody in Simar is involved with the project. Other units such as the geology school or faculty, and then we also have guests and visitors that were not included here. I didn't have the time to include everyone, but these are experts from other universities, from other groups, who make short visits and support us with identification, and this larger inventory keeps being generated. Here I didn't include the following because of the time constraint, but there's a whole series of companies that are partnered with the company, such as vessels, diving, lodging, restaurants, local businesses, where supplies needed for the project are purchased. So it provides a financial benefit to the region, and along with the development of these biodiversity projects. If we start going there and start holding activities, a small but important economy is established around the project and around biodiversity. Now, this is an example of sorts, so that you can see something that might be considered as a not so important organism. You can still extract information and start linking this to communities as well. We participated in social action activities such as talks at schools or marine festivals to help with the dissemination of these topics. And that would be all for me. Thank you. Thank you so much, Jeffrey. Coming up next, we have Dr. Odalis Cabrid, whose presentation is the marine uh, biodiversity of the ISEC Museum. She's a teacher at the biology faculty of the University of Costa Rica and is a member of the National Academy of Sciences. She's a researcher for the electronic microscopies research and of the Center for Investigation of Biodiversity and Tropical Biology of the University of Costa Rica. Her scientific interest is taxonomy and the Autocorellia systematics, taxonomic reviews, biodiversity studies for reefs, and also taxonomic studies of the mesotrophic and deep sea areas. She does taxonomic and ecologic studies of, of the mesotrophic and deep regions already mentioned. Good morning, everyone. Thank you for being present here. I'm going to share a little about the Zoology Museum. Monica Springer is the director, and Wendy Matamoros is the curator or collection manager. Next slide. Okay, the Zoology Museum is located in the bottom of the Zoology faculty in San Pedro, in the main faculty and main campus of the University of Costa Rica. Next slide. The Zoology Museum is the only museum in the country that holds marine collections. There are some other small museums that have a few collections, but this is the only one that is a museum as such. Next slide. The Zoology Museum has been declared by the government as the national repository for biodiversity, meaning that any organism that is identified in the country and that undergoes through any type of study has to be deposited in the Zoology Museum afterwards. Museums. Why are museums important? Museums in general, we can see these as security deposits where all of these species that are gathered and are going to be present for different type of work, or a lot of biodiversity is represented there. We can also see them as art galleries. These are truly spectacular. Many of the organisms that we have are spectacular, and we can speak about libraries, hardware stores, and pharmacies. A museum practically holds all of these functions together. Now we can also talk about why are collections important. There are many reasons why collections are important, and many of these reasons are because by having organisms available in the museums, there's less effort for gathering data or collection. So we're gonna have samples so that a group doesn't have to specifically go and sample these. They're going to be preserved for a long time. They're going to be preserved under adequate conditions as well. And we can minimize also the impact on the environment when collecting new collections, and it also maximizes the scientific value that these samples already hold. In the Zoology Museum, we have all of these marine groups, we have collections of all of these groups, and some other minor groups as well. Some of the most impressive or successful collections that we have is the ichthyology collection. This is the most complete collection of in all of Latin America. In this collection, there are more than 250,000 species, more than almost 20,000 lots, and there are freshwater and saltwater species, so we have both. Um, and we have fish from continental waters, from the islands as well. Another important collection, and one of the largest as well, is the mollusks collection. The curator for these collections is Yolanda Camacho, and there are about 38,000 taxonomic lots, about 250,000 species. There are bivalve, marine bivalves, more than 500 species, gastropods, about 250 species. Also, there are holotypes. A holotype is the organism that is the organism with which species is described. Here we have about 62 and 151 paratypes. The paratypes are the organisms that are next to the holotype when the species is described. This is very important and gives a lot of status to a museum to have holotypes and paratypes deposited. In the case of the fish collection, that is very large, we only have about four, because in the past the holotypes were not deposited. Only paratypes were deposited in the past, unfortunately. Oh, I also forgot to mention that this collection holds the collection that actually initially was started by Envio. So this collection moved on to be part of the
Otra de
zoology
las
museum
colecciones
collection.
muy grandes es la
Another one
colección
of
de
the
insectos
big collections
acuáticos.
is the marine
Sus insectos
insect collection.
acuáticos, esa
This
colección
marine
viene, la
insect,
hace Mónica
this
Springer,
collection
ella tiene
is
años de
has
trabajar
been
en
done by
eso.
Monica
Hay
Springer. She's
unas
been working on this for
95
many years. There are
familias,
about
unos 309
95 families
géneros,
and some
y
310
estos se ha enfocado
genders, and
mucho en,
this
digamos,
has
en
been
estudios
mostly
acuáticos y
focused
también en
in marine
estudios, digamos,
studies
de
and
impacto.
also
Hace muchos estudios
impact
donde
studies.
se
They do a lot of
revisan
studies where
lagunas o ríos, donde
lagoons
se
and
sacan
rivers
organismos
are
para ver
reviewed and
si el
where
río es
organisms are
puro o
extracted
no.
to determine
Entonces, todos
um,
esos organismos que
if the
se
waters are
recolectan
pure or
en ese
not.
tipo de
So all of the
estudios
organisms that are collected during
se
these type of
guardan también
studies
en el museo.
are
Por eso es que
also
es una
preserved in the
colección
museum.
bastante
So this is
grande.
why this is such a large
También,
collection.
bueno, la palabra es impacto
Also,
ambiental, se me
the,
olvidó.
the word is environmental impact. Sorry,
Y
I
también
forgot the term for a
se
moment.
usan para
But these are
monitoreo
also used for
y
monitoring purposes
se
too.
usan como referencia para
And
denuncias,
as a
porque
reference
cuando
for
hay una
filing
laguna o un
complaints,
río
because when there is a
contaminado, algunos
polluted river
organismos
or
pueden
lagoon,
decir, o
some
sea,
organisms
pueden ser testigos
can
de que ahí hay contaminación.
attest
Y
that there
también
is
ahí en el museo
pollution
se
present,
registra eso.
and these are also
Siguiente.
deposited in the museum.
Esa es la colección de
The
Nidaria.
Nidaria
Sí, obviamente iba a poner la
collection,
mía, ¿verdad?
definitely I was
Pero
going
es
to
una de las
include
colecciones
my
más
collection, but
completas
this is
que
one of the
hay,
most complete
incluso en
collections
Latinoamérica.
in all of Latin America,
Tenemos
even.
especímenes, una gran cantidad
They
de
have a
especímenes,
large amount of
unos
species, some
40.005 especímenes. No tenemos solo especímenes de
40,005
Costa Rica,
species, not
también
only
tenemos
Costa
de El
Rican species,
Salvador,
but also from El
de la
Salvador,
colega, de nuestra colega
from our very
Joana
dear
Segovia,
colleague
y
Joanna
tenemos de
Segovia,
Panamá una
and
colección muy
also from
grande.
Panama, a very large collection from Panama
Entonces,
as well.
es una de las colecciones más grandes y
This
tenemos
is
más
one
de
of the
30
biggest
holotipos
collections, and we have
y
more than 30 holotypes
226
and
paratipos.
226
Siguiente.
paratypes. Next.
Bueno, las facilidades que tenemos en el
Okay,
museo.
so the
Tenemos una colección
characteristics of the
húmeda y
museum
una
is that we
colección
have
seca.
a moist and a
Porque hay
dry
organismos
collection, because
que
there
tienen de
are
las dos.
organisms
Tenemos
that have
organismos en
both.
alcohol
We have
preservados en etanol
organisms
y tenemos
preserved in
organismos que
ethanol
se preservan
and
secos.
others that are
En este
preserved
museo,
in dry
y además,
conditions.
3
In the
congeladores a
museum, we have
34 grados
3
centígrados,
freezers at
un
4
congelador de
degrees
-20,
Celsius, a
tenemos un
freezer at minus 20
laboratorio
Celsius. We
húmedo
have
y
a moist
espacios para
laboratory,
trabajar y
also
equipo
space
óptico
for
para
work, and
poder trabajar ahí.
optical team
El espacio
equipment
está
to work with.
disponible para
The space
visitantes
is
cuando quieren
open
llegar
for
a ver alguna
visits,
colección o a revisarla.
for whenever
Siguiente.
somebody wants to make a visit or review some type of collection.
Entonces,
Next.
digamos, redondeando números, hay
Now,
como
rounding up
2.5
numbers, there are about
millones de
2.5 million
especímenes catalogados.
species that have
Hay
been
muchos otros que
cataloged.
todavía
There are
no
many
tenemos
others that we
catalogados. Hay
still
parte de
do not
los
have
especímenes del proyecto
cataloged.
Biomar que habló
Part of
Jorge,
the Biomar
que todavía no están
species
catalogados, no entraron
are
en
not
nuestro
still
catálogo
cataloged,
oficialmente,
have
pero
not
bueno,
gone
están
into our official
procesando
catalog yet,
unos
but they're being
150
processed.
lotes en proceso.
About
Además,
150
tenemos una
lots in
gran cantidad de
process. Besides, we
vouchers que
have
no
a
se han
large amount of vouchers that
catalogados. Los
have
vouchers
not been
son
cataloged.
especímenes
These
que se
vouchers are
recolectan cuando se
species
hace un
that
estudio,
are collected
pero digamos que son
when
muy
a
limitados,
study is
casi
made, but
todos.
let's say
Una
that they're
misma
all
especie,
kind of the same
¿verdad?
thing, right?
Y
They're all
hay
from the
una
same
obligación
species,
cuando
and
se
there
hace ese
is an
tipo de estudios
obligation
incluso
when
de
this type of
depositarlos en
study
el
is done
museo.
to deposit these in the museum as well.
Tenemos organismos
We
preservados
have
en general en alcohol al
organisms that are
70%.
preserved in
Algunos han sido
70%
fijados
ethanol.
previamente en
Some have been
formalina, se lavan y después se
previously
pasan a alcohol desinfectante.
fixed in formaldehyde.
En
These
general,
are
lo que
washed
hacemos
and
es
then
fijar
moved
ahora
on
como
to
el
70% ethanol.
95% o
What
al
we are
menos
fixing is
una
at least
fracción de los
95% now, or at
especímenes,
least a
una parte
fraction
de
of the
algún animal para
species
tener
that
una muestra para
have
trabajo
a
molecular.
sample for molecular work.
Bueno, también hay una colección seca
There's
de
also
conchas, de
a
corales,
dry
de equinodermos y
of
también
corals,
insectos
of
acuáticos.
shells,
Siguiente.
and echinoderms and aquatic insects as
Entonces,
well.
las colecciones que tenemos
So
de
the
marinas y
collect the
agua dulce
marine
también
and freshwater collections that we
son
have
colecciones de investigación.
are collections for
Ahí
research.
se sacan publicaciones científicas sobre
Scientific
biodiversidad,
publications are
taxonomía
made
sistemática.
from
Y además
taxonomy
se tienen todos
and
estos
ecology, and
vouchers
besides, we
de
have all
trabajos
of these
para genética,
vouchers for
monitoreo y
genetic
taxonomía. Siguiente.
monitoring and taxonomy work. Next slide.
Antes de esto, quiero decirles que el
Before
museo
these, I'd like to say that the Zoology Museum is not an exhibition museum. This is why I didn't mention it. This is a research and investigation museum. It's basically for investigation. So we have many species described by the museum's curators, many others that have been described by other collaborators, but in general, for fish, we have 76 species described by curators. In corals, 45, mollusks, 9, crustaceans 6, including
y
a new
muchas otras,
gender and
como les dije, de otros colaboradores.
many others
Hay
from
muchas
other
especies en este
collaborators. As mentioned,
momento
there
que
are many
estamos
species at the
trabajando que
moment that we're
probablemente van
currently
a ser
working on
nuevas,
that are probably going to be
especialmente en
new,
las colecciones de
particularly
moluscos
the mollusks
y las
collection
de
and
nidarios.
the
Bien,
Nidaria one as well.
entonces, ¿cuál es el uso de esas
So,
colecciones?
what
Ya
is the
les dije
use
un
of these
poco.
collections? I've mentioned
Nosotros
this a
hacemos
little
también
already.
intercambio de
We
material
do
con otras
material
instituciones
exchanges with
científicas.
other
Por
scientific
ejemplo,
institutions. For
otro
example,
científico nos
another
pide
museum
que
or
quiere
another
rescatar
scientific
material, entonces se lo
requests a
enviamos o
review of
nosotros
X or Y
recibimos de
material,
otro
so we
museo
send it, or
material
we
en préstamo
receive a material
para
from
poder revisar
other
algunas colecciones.
museums as a
Aquí
loan
a veces
through.
hemos tenido problemas
So
con los
we've had
permisos
issues here with
después para
permits
devolver, pero son
and the
cosas que ya
return
estamos trabajando.
um, items
Eh,
back, but these are things that we're working on
Después,
already.
los especímenes depositados
Then
también
the
sirven para los
deposited
cursos de la
species are
carrera de
also
biología
useful
y también
for
para artistas que vienen
classes
al museo a hacer
in the
dibujos, como
biology
esta estrella que tal vez
faculty,
la
and also
reconocen,
artists that
algunos
do
saben qué
studies.
billete está,
Maybe
probablemente no
this
se han
this
fijado, ¿se entiende?
sea star is
Alta
probably
observación.
recognizable.
Como les digo, no somos
It's
un
actually
museo
in one of our bills.
de exhibición,
As I
pero
said, this is not an
participamos en
exhibition museum, but
exhibiciones que
we
hay de museos
participate
en Costa Rica.
in other exhibitions
Tenemos
from other
pequeños
museums in
stands
Costa
donde
Rica.
llegan
We set up little
estudiantes
stands
y
where
se les enseña
students
alguna
will
parte
set
de
up,
nuestra
and,
colección.
a small
Bien,
part of our collection is shown to them. Next slide.
entonces en general,
So,
estos son, ya se han dicho,
in
la
general, I
importancia de
think
las
that I've mentioned
colecciones
these already. The
para
importance
estudios
of
de
collections.
taxonomía y
These are important
sistemática,
for
morfología,
systematic and
anatomía,
taxonomy studies,
genéticos,
for
estudiar
morphology
la
and
distribución de
anatomy, genetic
organismos,
studies,
estudios
distribution of organisms,
ecológicos,
ecological
estudios sobre
studies,
enfermedades de
studies
animales,
on animal
también sirven
diseases.
de colecciones de
These are
referencia.
also
Estamos
important as
tratando
reference
de poner en
collections. We're
línea
trying
todas
to
estas colecciones
put
una vez
all
que
of the
tengamos todo
collection online
catalogado
and
para
want
que
to see
esté
everything
disponible
cataloged
para
so
todas
that
las
it is
personas que puedan
available for
ingresar.
everyone
Esto es un proceso
and for
caro,
people
lento,
to
pero
be
estamos
able to
trabajando en eso
see these
y
online.
pronto
It's a
se
very
podrá,
expensive
como les
process, but
dije,
we're working
los
on it and it's
organismos
going to
que
be available
tenemos son
soon. As I
testigos,
said,
evidencia
the
real
organisms that we have are
y
witnesses,
también
evidence
podemos
of
dar
the
una,
environment, and we can
todos estos
also
organismos sirven como de
all of
línea
these
de base
organisms
para
work as a
información en las
baseline for
comunidades y
information
también nosotros
in the
apoyamos
communities, and
exposiciones
we
temporales
also
o permanentes.
support either permanent or
Esa es
temporary
la última.
expositions as well. I believe this might be the last
Siguiente,
slide. Is
tiene la
there
palabra.
next?
Bien. Gracias.
Oh, okay. Well, that's it. Thank you.
Bueno, para mí es un gusto presentar a la doctora
Okay, so to
Cindy
me, it's a
Fernández.
pleasure to present Dr.
Cindy va a hablarles
Cindy
ahora
Fernandez.
sobre
Cindy
la
is going to
biodiversidad
present
marina
about marine
en la
biodiversity
Universidad de Costa Rica.
in the University of Costa Rica
Cindy
Herbiary.
es máster en biología de la
She's a
Universidad de
master in
Costa Rica,
biology,
tiene un
graduated from
doctorado
the
en
University of Costa Rica. She
biología
has a PhD in
costera
marine
del
and
estado
coastal biology
California
from the Baja
Sur.
California
Es profesora
Autonomous
asociada en
University.
biología
She's in a
marina
partnered
de la Universidad de Costa
teacher
Rica
at the
y
University of Costa
presidenta de la
Rica,
Asociación
biology faculty,
Biológica de
and also
Centroamérica
president of
y
the
del Caribe.
Ecology
Es
Association
investigadora
of
del
Central
Centro de
America and the Caribbean.
Investigación en
She
Ciencias del Mar y
heads
Tecnología,
the
CIMAR, y
CIMAR
del Centro de
and
Investigación
also
en
the Research
Biodiversidad
Center for
y Geografía
Biodiversity and
Tropical.
Tropical
Su interés
Biodiversity,
científico es la
CIBED.
taxonomía
She's interested in
sistemática y
systematic
ecología de las
taxonomy,
macroalgas de Costa Rica y
macroalgae
Centroamérica.
taxonomy of Costa
Recientemente
Rica
ha
and Central
incursionado
America.
en el
She
programa de
has recently
desarrollo
incurred
de
in the
los
development of
potenciales usos de las
potential
macroalgas.
uses of
Su
macroalgae.
formación también es importante en la formación de
Her preparation is
profesionales
also important
de la
in the
biología.
preparation of professionals in the field of biology as well.
Bueno, esta presentación es un poco la continuación
So this
de la
presentation is a
presentación de
continuation of Odalisca's
las
presentation.
colecciones del herbario,
The collections at the
museo,
Herbiary and the museum, both the
Universidad
Herbiary and the museum, both belong to
de
the Research Center of Biodiversity and Tropical Biology of the university.
una cosa
And
importante
something
en las
important about
la interacción
the
entre
interaction
instituciones
between research centers is that
tanto el CIMAR como el
CIMAR and CIBED
trabajan
both work
para
together
esclarecer
in
la
order to
biodiversidad.
clarify
Es como
our country's biodiversity. So it's a very particular bond between the research centers that takes place within the university. This work is I I'm part of the work, but I also have a whole bunch of students working with me. And currently, these names that we see on screen: Mariana, Monica, Laura, Jose, and Jose Manuel. These are students who have worked but recently many others too. But they have recently worked on the herbary and these collections themselves. The University of Costa Rica Herbary, with the USJ acronym, was founded in 1931, and it holds approximately some 115,000 species. Mainly, it started as a terrestrial plants herbary, and it started including some kelps and funguses. Currently, the national legislation establishes that if you collect organisms, you can deposit them in the National Museum with the CR acronym or in the university's herbary.
So this is a vision, a visual of how the herb collection looks and the different specimens that we have. The marine collections represent approximately 3% of that 1,000 and something specimens that we have registered. And this marine collection has two components. First, which is the marine algae and the marine grass, and then marine pastures, and that covers plenty of the national collection. And the three groups are represented. We have the green, red, and yellow algae. The collection of macroalgae, marine macroalgae collection, was founded by Professor Ricardo Soto till the 90s, and he worked with a collection specifically from the Caribbean. He worked till the 90s, and then we had a German professor that came, and she really increased the collection, and we have a very big collection of macro seaweed from the Caribbean, and that was thankful that was thanks to these two investigators. I also began and I started to collect more from the Pacific, so that's where the Pacific region and that portion grew in the collection. In total, we have 155 collectors of macro seaweed, and they have collected those specimens and placed that in the herbary. And we have more than 350 sites that are represented in the herbary. We have around 70% of those specimens from Costa Rica, but 30% is from other countries. And we have a very good representation from Central America here in this herbary. This is a graph that was set up in 2021 by my student Jose Manuel Picado, and as you can see, the light colors are where there is a strong effort for the sampling and collecting. And we have some areas in the Pacific near the Nicoya Gulf. And so what we're doing is we're moving this again. We're having new information to run these analysis again, and it's very possible that the North, thanks to Biomar project, also has a good collection as well. So we're focusing on the darker blue areas in the Pacific, which is where we're having more. And we have some Caño Island and some other areas in the South Pacific as well. The collection of macro seaweed are divided in two, dry and moist. And we have 3,474 in the dry collection, and 900 is about to enter. And then there's a moist collection that's always in alcohol, that always helps us to be able to see the marine organisms kind of in a for fresher way because they are usually dried out and kind of laid out, but the moist collection kind of helps us to see how that would actually be in a more real way. And then we have the macro seaweed. We have that around 4% is diatomeas. We have another one, another collection. It's cyanobacterias. And we have a strong effort to see what they are, what we have in our country, and that's used in pharmacy, and it's very important. We have the pardas on this graph, and then we also have the green seaweed. Um, we have then the brown seaweed, green seaweed, and red seaweed, and that's all in the graph there. And to finalize, the collection of marine pastures has 74 dry specimens approximately. And one of the gender that it's better represented is the Halophia, Habdul, Tulasia, and Cypnodium. So it's a small collection, but it's very important. So that's one of the important collections that we have. Okay, so thank you for that. And now we will have the Yolanda Camacho professor. She will be presenting Protecting Our Marine Biodiversity, the impact of the collection of shells by tourists in Costa Rica. She's licensed from the biology school, also from the University of San Francisco in California, and a doctorate in the University of Costa Rica. And she works in bio and also as a teacher for the biology school for the University of Costa Rica. She's also inscribed in the investigation department for this university, CIBED, CIMAR, and CIBID. Yes, thank you. Okay, so yes, thank you. Well, this is an informative presentation to raise awareness and to let you all know of different challenges that the international airport is facing for some years now, and that's related to the impact of the recollection of shells. So the content of what we will be presenting: biodiversity, tourism laws, and we have content also. Okay, so and also seizures. Okay, so our country is very benefited from the biodiversity. We have 5% of the biodiversity of the planet. We have national parks, protected areas. We have beaches, two beautiful coasts, different volcanoes, forests, and all of that makes Costa Rica one of the countries most visited in the area. And Costa Rica is stable and has very nice, welcoming people. And tourism is our first means of income in this country. Last year, in only 1 year, in 2023, Costa Rica had a visit of 1,700,000 tourists in the Juan Santa Maria Airport. And in Daniel Uber, we had many more, and that gives a total of 2.5 million people per year, with many income coming in related to tourism. We have the Ley of Conservation for Wildlife, and that is 7317 from CINAC, and this law establishes that it is prohibited to collect, extract, and hold wildlife, all of the organisms, parts, products, and other areas
se pueden implementar
that may
sanciones
be
hasta de 3 años
derivatives.
base para aquella
And
persona que infrinja
the
esta ley.
sanction is of
Las
to
conchas y los
30
corales cumplen
salaries.
varios roles
So,
importantes en el ecosistema.
shells and coral reefs
Por ejemplo, la
have an important
descomposición de las conchas hace
objective
que
in
el
our
carbonato
country.
de calcio se vaya
For
se
example,
disuelva en el agua,
the dissolution
que la
of
concha del océano
shells
y también
in,
pues
affects
hay
the
más
chemistry of the
micronutrientes para
ocean
otra gran cantidad de
and
organismos
breaks down
marinos.
nutrients for
También
other
las conchas
marine
sirven de hábitat y
organisms.
refugio para una
These
gran
shells
variedad de organismos.
also
Por
are
ejemplo,
the
bueno, estos
habitat and
organismos las usan
refuge for
como
many
protección y
microorganisms.
también las usan para
So many are
poder
used
defenderse
for
o poder
protection
protegerse
by
cuando hay
several
condiciones ambientales
species,
adversas. Tal es el
and when
caso
there
de que
are
tenemos
some sort of
algunas especies
perhaps
de
danger, and we
cangrejos
have
centrales que
some
las usan, usan
crabs, for
conchas
example,
y
that
vacías para
use
protegerse.
empty
También hay
shells.
pulpos que las utilizan y otras
And there are
especies
also some
peces.
octopuses that
Bien,
use that in the same way, and
el
also
hecho
some
de que
other
haya
fish
conchas y
species and
corales
different
en el
fishes.
lecho marino
So the fact that
hace
there
que
is
la diversidad biológica
shells and
aumente. ¿Por
reefs
qué? Porque hay
in the
muchas
marine
algas y
floor
muchos biomas,
makes the
microambientes, y eso
ecosystem,
aumenta la riqueza biológica, la
it
complejidad
makes
del
it
ecosistema.
increase and it affects the
También las
complexity
conchas,
of the
el que haya
ecosystem.
conchas en el
The
sustrato
fact
lo que
that
hace
there are
es estabilizar esos
shells
sustratos para que otras
on the
especies crezcan,
substracts
como las algas y los
can
pasos
also
marinos.
grow maybe it's like
Siguiente.
seaweed or pastures.
Un estudio reciente
A
muestra
recent
que en
study
la costa
showed
caribe de Costa
that
Rica
in
tenemos
the
un
Caribbean
13%
coast we have a
ya de erosión costera.
30% of erosion,
Para que ustedes
coastal erosion.
sepan, las
So you
conchas
can
y los
kind of
corales
understand
actúan como barreras
shells
protectoras en el ecosistema.
and
Las grandes barreras
coral
coralinas, las
reefs
barreras
serve
telúricas,
as a
los
border,
serpentones, etcétera,
as a
lo que
protective
hacen es proteger
border,
las costas de la o controlan la erosión.
as a
Por ejemplo, lo que
barrier
hacen
that
cuando llegan
also
las
controls
olas
erosion
al haber estas
because
barreras se
the
distribuye la
waves
energía de
hit
las olas y entonces se
these
protegen las costas.
barriers,
Si
and
nosotros
then
removemos
therefore that will
este material
take away
de las
energy
costas,
from that wave, and then the
eventualmente
coast
vamos
will
a
also be
tener que
protected.
enfrentar
If
una
we
mayor
remove that,
erosión
we will also
costera.
have to face coastal
Siguiente.
erosion.
Desafortunadamente, con la
Unfortunately,
incrementación en el turismo, se han
as
visto
tourism has increased,
más conchas incautadas en los
we
aeropuertos.
have seen
El turista
more
quiere estas conchas
shells
porque quiere llevarse un
taken
souvenir de Costa Rica, lamentablemente.
in the airport.
Y
So
bueno,
tourists will
hay
pick
aumento
those shells up
significativo
because
en
they
las
want to take
incautaciones de
a
las
souvenir.
conchas en los meses de alta visitación de los
And
turistas.
there is a removal of these shells from the
Estos son los
beaches
números de las
where
incautaciones
tourism
en el
it's in
aeropuerto
its
a
peak
nivel mundial
season.
entre el 2021 y el 2023.
So when have
En
we
2021 se
seized that in
incautó
the
1000
international
kilogramos y en
airport?
2022
In
1000 kilogramos, un
2021,
poquito más,
it was
y en
around
2023
1,000
1900 kilogramos. Ahora vamos a ver lo que
kilograms.
pasa
In
con el cuarzo
2023,
submarino. Siguiente.
more than
2021 se
around
incautó
1,800
900
kilograms.
2022 2300 kilogramos y en el
2022
2023 2900 kilogramos.
2022
Esto
was
nos da un
2.3
gran
kilograms,
total.
2023 2.9 kilograms. So we can
Toneladas de
see
material
the total
incautado
of that
en
would
3
be
años.
around 10
Esta cifra es
tons.
abrumadora.
That is a lot
Y
in
como
just
pueden
3
ustedes ver
years, 10
en el
tons.
gráfico,
And
hay
as you
una
can see from the
tendencia
graph, there is
hacia
a
un aumento
tendency
de estas
for that
incautaciones.
to increase, for people to seize those shells and to
¿Qué es
take
lo que
them.
los turistas están
So what are
llevándose de
tourists
nuestras
doing?
playas?
What are they taking from our
La
beaches?
lista es bastante grande.
The list
Principalmente
is quite
conchas y
large.
corales.
Mainly
Entre las
shells,
cuales podemos
corals.
encontrar bivalvos y gastrópodos,
We can
corajes,
find
corales
some
que están
bivalves,
protegidos por ley,
gastropods,
CITES.
crustaceous,
Tenemos también
fishes,
crustáceos y peces secos, equinodermos,
dry
galletas de mar
crustaceous,
y erizos.
fossils,
También
rocks,
se está hallando un
sands,
alto porcentaje en rocas fósiles, entre las rocas están
seeds,
cuarzos,
dry wood,
muchas botellas de arena,
some a
semillas,
lot of bottles of
María Serra,
sand.
y bueno, otras cosas. Siguiente. Este es un
Next,
ejemplo del material incautado.
this
Tuvimos
is an
la suerte de
example
visitar,
of
hacer una
the
visita
different
al
materials that
concentrario
were
de hace un par de
seized.
años. Y estas son
Um,
algunas de las fotos de lo que
we had a
encontramos.
visit to the airport some years ago, and these
Encontramos
are the things
desde
we found.
pequeños, la gente recolecta desde un puñito
People
de
would take
conchas,
from
¿verdad?
a small portion
Hasta
of shells to
bolsas con kilos de
bags
conchas,
of kilos,
incluso me han dicho que hay
kilograms,
hasta kilogramos, más de un kilogramo de
maybe
conchas.
even
Entonces,
more than
por
one
lo general se está
kilogram of
recolectando el
shells.
material, las conchas vacías, pero
So
también
generally, the shells
pueden haber
are
recolectado también los
empty, but
animales vivos.
sometimes they'll take the live organism inside the shell
Este
as
es el
well.
destino final del material
This is
incautado
the final
actualmente.
destiny of
Todas las
the seized
conchas que
material
están
currently.
incautadas en el
All
Juan
of the
Santa María
shells
son
that have been
almacenadas en dos
seized
contenedores
are
de 1.000
stored
kilogramos cada
in
uno y
two
en sacos
containers of
de
1,000 kilograms each
jute
and
grueso.
in
Y
bags.
después
And
se
then
abre un hueco
a
en
hole is
las
opened
mismas inmediaciones del aeropuerto
in the
y
same
se entierran
airport,
las conchas.
and then they just
Todas las conchas ahí van a
bury
parar,
that there.
son
That's where
basura.
all the shells will land. They're basically trash.
¿Qué es lo que se ha hecho hasta
What
ahora
has been
para
done
mitigar y
so
educar a la
far
gente?
to mitigate
Han
and
habido
educate people?
muchísimas campañas de sensibilización a
There
la
have been
población
a
con
lot of
material
campaigns
audiovisual,
to raise
infografías,
awareness for
prensa,
the
televisión,
population,
comunicados en redes sociales, en fin.
TV, audiovisual
Sin
material,
embargo, en este momento
campaign,
no hay
um,
inspectores de
through the
vida
press,
silvestre en el Juan Santa María,
social network,
en el otro aeropuerto tampoco,
but there
haciendo
isn't
las incautaciones.
submarine
Y faltan
export in
formularios
the
que
airports,
recopilen
in any of the
información
two
del
airports,
material incautado.
where they
No,
can
el
really
turista básicamente
use that and
sale del
handle
país
that. So basically,
sin
the
ninguna,
tourist leaves the country without
haber llenado ni siquiera un
really
formulario
even
o
having
haber
filled
recibido
out a
una sanción.
form or received
En este
any type of
momento
sanction.
hay la
Right
asesoría
now,
legal del
between
SENAC
the
y la
legal
Fiscalía
assessment
Ambiental
between
están
SENAC
haciendo un análisis de la
and the
normativa vigente
environmental
para ver
ministry
si se
are
pueden
making an
implementar las
analysis to
sanciones.
see if there can be a sanction placed.
Estos son ejemplos de las notas de empresa que han
These
salido
are
por
some of
medios de
the
comunicación,
publications through the
tanto del Gobierno
press
Nacional como
that have been put out
La Nación, CR Hoy,
through different newspapers, La Nación,
Times,
CRO,
y algunas
Tico
infografías
Times,
que se
and
han
some
visto
infographics
en
that
la campaña de Visual Hero en el aeropuerto de
that is also called a campaign that's called Be a Shell Hero.
Una concha sí
And a
importa.
shell does
Cuando
matter.
estamos tomando una concha
When we obtain a shell from the ecosystem, we are stealing the opportunity, we are taking the opportunity for different organisms to obtain the nutrients that those shells have and help them to, which helps so that they can create their own homes. And it's really provoking a irreversible effect in the environment. We need to intensify campaigns for awareness, for national and international tourism. Inspectors of this wildlife in airports would be very important, and also enforce the wildlife law through sanctions. So this is a call to action. Many of us, our families, our in our homes, our friends, we have most likely have gone to beaches and taken shells, and we just take that into our home, and then they end as decoration. And this is something we need to change. Definitely, we need to begin to change this, and instead of bringing the shells home, we should take pictures, not the shells. The numbers that I presented are the numbers that we know that are registered for tourists. We don't have any idea of how many, of what the impact is national tourism has in these organisms. And I want to thank SINAC, to the ACG, IARES, the conservation area, to help me to gather this information and to allow access to the information that was presented. Thank you, Yolanda. Thank you, Yolanda. And now we will have Dr. Kayla Rojas with the studies biodiversity and different microorganisms in different environments. Kayla has a doctorate um, in different biology subjects from different universities, also from Marburg, Germany, and from the Leising Institute in aquatic energy. And he's also a teacher at the university at the moment. And his investigation has to do with the study of biodiversity, genetic, and of microorganisms in different natural ecosystems.
Good morning. Thank you for the invitation. Different than my colleagues, I'm going to present the results of the different research works, but that represented the large amount of marine organisms that we have. I'm going to start thanking Mr. Jorge, Mr. Jeffrey, Maria Jesus, Melanie, Frank, Yvonne, and Stephanie Solano. Next slide, I'm going to talk about results, some very fresh of this just last weekend, but I really like to show the great amount of organisms that we have. First is the bacteria and fungi biodiversity at a mangrove in ACG as part of the group that is worked on with Biomar. Then I will speak about fungi in deep waters of the Pacific Ocean. And this is part of a crazier idea is the plasmidome studies in deep waters worldwide. To begin with, in the North Pacific area, there was a mangrove that before, but well, it was a mangrove, then it was a salt extraction point, and the ACG started the restoration process there. And the restoration process it was to develop channels so that the seawater could flow in and making the primary and secondary channels, and then planting some mangrove species such as Avicennia rhizophora. We have another mangrove that is used as a control, which is the one in Hunquilla. And we sampled in different areas to review the variety of microorganisms and fungi, and also to see the state of recovery of the mangroves so far and see how things have played out. Next slide, please. Here's where I'm going to use my umbrella to present some results. So here is the salt extraction company control. So I'm not going to go into detail for each of the groups, but you can see that these is very different to the control group, and from the mangrove and the restoration area, this part is basically just salt. And from microbiology perspective, is very interesting because wonderful. How does this? In this part, we found very interesting microorganisms, so groups of bacteria that are extremophilic, not only bacteria but also um, archaea, which is a very interesting part of the tree of life. But what I would like to highlight is that if you see um, the mangrove in terms of restoration is very similar to the one in restoration is very similar to the one in control. So we can see that the actions that SINAC has taken is having an effect just by reviewing the microorganisms populations. There are just a few groups here that are differentiated. I, with time, have learned to see that ecosystems are species, and in terms of bacteria, they're also functions, or in terms of microorganisms. So we have microorganisms with very interesting functions. And here, for example, I have a group that a while back I thought that should be included, and we fished them out. They're called sulfobacteria. It's a group that produces what it's called cable bacteria in English. It produces an electric cable that produces electricity on its own, and there are people using them for renewable batteries for cell phones using bacteria. These species is found in the mangrove. The other thing is that I've been studying biodiversity all around the world and throughout different ecosystems for many years, and I had never seen in these mangroves we found 40,502 ASVs, which is an equivalent to species. A while back, I made a study in a cavern and I found 16,000, and that was already a high amount. For example, in the human microbiome, you found 2,000, 3,000, 40,000 is one of the highest numbers. I can't even process it in an Excel spreadsheet that has 15,000 columns, right? So it's something tremendously interesting in interesting metabolites. Here, for example, when we see the richness, we see, and in fact, this is part one. This is the closest one to the ocean, and part three are the channels that are a little bit further away. So one can see how the richness of species uh, decreases, but also with great concentration. If you see Shannon indexes, for those who are familiarized with it, if you see an index of 8, I had never seen one. Normally, an index of 4 was already very impressive. Another thing is that for those who are familiarized with ecology, one always sees some symmetry in the composition of communities. Here, there are relatively symmetric. So, what I'd like to sell here is this: the mangrove is really interesting, and this interaction in terms of species diversity is really remarkable. We are seeing it here just with with DNA, but if one started isolating, then the amount of new things that one could find would truly be fascinating. Next slide. However, with fungi, the same thing is not happening. This is the control site, and you can see that it's very well differentiated from the mangrove that is in restoration, or the site where the pure salt was located. In here we can see that the fungi population are not being restored at the same speed as bacteria populations are, which is why it's interesting to have two different systems to measure, to have bacteria plus fungi as supplement. But another interesting part is that in these areas of recovery and in the salt extraction point, we've found many types of yeasts that I suppose have very special adaptations in order to survive to the high amounts of salt. Sometimes they have glycerol accumulations or things like that in order to survive, or thickenings in the membranes. So, from a metabological perspective, it's truly interesting, and also the numbers are much higher than in other ecosystems. Next slide, please. Now, the second story I'd like to narrate is about the fungi communities in deep water, so I'm not going to emphasize in the introduction too much. Mr. Jorge already presented some information, but I would just like to remind everyone that for a fungi that lives in deep water, it has the sustained total darkness, hydrostatic, for example, the 3 kilometers of water, and the weight and resistance in order to live in those conditions, low temperatures, um, low amounts of oxygen and the nutrients available. So it has to have special adaptations in order to live in these ecosystems. Next slide, please. A few years back, and just to contextualize why fungi in these ecosystems, a few years back we published the idea that nobody questions the fact that fungi are important in terrestrial ecosystems and in agriculture, but I don't know why we scientists are just forgetting about fungi in the ocean, or the old belief that there were no fungi in the ocean. But what we believe is that they have equivalent ecological functions, just as in terrestrial ecosystems as well. Here, Mr. Jorge has participated in numerous expeditions to explore fungi in deep water sediments, and I'm going to summarize. In a depth of 400 to 3,500 meters, we found 787 different types of fungi. For each site that was sampled, there were approximately 75 fungi per sampling point, and we found that these species, this is a type of yeast, was the most common in the majority of the sites, and that's particularly with a more of a muddy landscape, and the amount of space that the deep water represents in the country. To me, this would be the most abundant organism in all the country. We have tried to fish it out, to cultivate it. We haven't been able, but we found two clusters where yeasts are found in the more muddy areas, and they're in the more sandy areas. There are other types of fungi which are more filamented types of fungi. Next slide, please. Now, what we wanted to do later was okay, let's try to fish out and cultivate these fungi. And one would think, okay, so they live at 3,500 meters of depth in total darkness. They're not going to grow in the normal atmospheric conditions of and temperature that we live in. However, we were able to cultivate 54 strains belonging to 36 species and 8 different orders. And that also represents the first strain collection of fungi, and we're trying to have the conditions to cultivate them. And additionally to these, in order to try to understand what sort of activities it might have, so we did some enzymatic assays, and at least from these two, we've uh, uh, we, this could indicate that this fungi have the capacity of degrading ketene from other insects, and it's incorporated into their system. We found these two type of fungi that are representative, and we're currently sequencing this one. And this is a fungi that can behave as a yeast or as a filamented type of fungi, and it's also of a deep dark color, so it produces some type of melanin. This type of melanin works as sort of a molecule that allows it to survive the high osmotic stress or pressure. It also it's also very interesting from the genome perspective. So I would just like to point out that these are just a few selected bullet points from this big potential found in 92% of the country, and fungi that are usually left aside. And I'd just like to remember everyone these are very interesting too. Thirdly, the third story is more related to a bit of a crazier idea. I'm not going to expand on it too much, but I'm going to mention that when one speaks about genetics, we found that normally microorganisms have one chromosome, and they also have extra chromosomatic elements, other mobile elements as well. But the wild idea is that we are proposing that these genetic elements could be considered life, and if these are life, we can do taxonomy, ecology, evolution on these. But well, this is a different story for another day. But we wanted to consider this, and we try to analyze it in a very unique ecosystem, such as deep water ecosystems, and we wanted to analyze the uh, the plasmidome populations and the plasmidome populations in deep water worldwide. So for these, we used, or well, we would develop some bioinformatic pipelines along with Oslo University and here with a SENAC colleague. And from the samples and all the data, there are some international expeditions. We took their data, we annexed everything that was related to chromosomes, and we annexed plasmidomes as well, and did a study of plasmidomes worldwide. Some very interesting things. We had two different depth profiles from 200 to 1,000 meters, and from 2,400 to 4,000 meters. We found two populations. I supposed that we were going to find plasmidomes in deep water ecosystems that were very deteriorated. We've found, for example, in Rio Virilla, that a single bacteria has 13 resistant genes, and they're all found in the same plasmidome, which is very disastrous. I thought that we were going to see this in deep waters as well, but luckily we only found 7 resistance genes in all of these very large databases, and all only in the 270 to 1,000 meters layer. So one could say, okay, luckily we haven't deteriorated the other layers that much. The deeper layer of 2,400 to 4,000 meters, we at least didn't find any resistant genetics, but we found very interesting things like such as genes related to metabolizing heavy metals, for example, or genes related to the capacity of surviving in these ecosystems. Next slide, please. Um, in general overview, um, we've found that plasmidomes populations have behavior similar to a chromosomic or bacteria population or any other organism. And we also found differences between the upper layers and the lower layers, and that populations are clearly distinguished one from the other. We generally found that the upper layers have higher amounts of plasmidomic species or associated taxonomy, that the size of the plasmidomes are also bigger, a higher amount of resistant genes in metabolic lives. This is again just evidence of what one can find in the ocean worldwide, and we continue accruing information about different ecosystems to propose that these mobile genetic elements could be a new type of life, but we have to expand the classic definition of what we consider as a living being or not. Thank you. Thank you so much, Kaylor. In order to wrap up with our panel, we have Dr. Maria Sandoval. She is an associate teacher of the geology faculty at the University of Costa Rica. She has a PhD in soil sciences in Usana University in Sweden, obtained in 2015. She's a specialist in micropaleontology, particularly in radiolaries, and their projects are focused on paleoceanography of sediments and deep oceanic rocks in the Pacific and the Caribbean, with ages from the Cretaceous to the Holocenic. Thank you for the invitation. I am going to talk about studies that we've been doing in the geology school. Part of these studies are thanks to Jorge, who has given us a lot of work here at the geology faculty, and precisely the radiolaries and foraminiferous studies, and those of us who work these species have been developed mainly at the geology faculty and biology school, at least the University of Costa Rica, are a little bit set aside. They're a little bit abandoned, as Keller mentioned. So in collaboration with Jorge, we have worked on these. So Alexis Rodriguez, who's also a student, who's also doing his master's degree in biology. Viana Gamboa, who's also a doctor specialized in foraminifera, and her students, also Mr. Jorge included. So what are these? Foraminifera and radiolaries are organisms that we could say are mainly oceanic marine. In the case of radiolaries, they are exclusively marine and planktonic. In the case of foraminifera, these are also organisms that are part of plankton, but they're also benthonic organisms. Um, the differences at a general level is that these are microorganisms, microzooplankton. You can see here the scale. The scale is of 300 micrometers. They could be a little bit larger or smaller. They're made of calcium bicarbonate, and radiolaries are made of biogenic opalum. This is part of their characteristics. For example, in the Eastern Pacific, they are very abundant, and in plankton networks and sediments, they are very abundant, and we are in charge of identifying and counting. Previous slide for a moment, please. I just wanted to make a comparison towards the recent studies and geologic registry. As I said, these organisms at a geological level, they are very important, mainly in geology. They're always used in the oldest pieces of rock of the country. These are about 180 million years old, but they have lived. These organisms have lived for the past 500 million years to the present. So these studies are very particular and very well developed in the geological registry by using these organisms for adaptation and whatnot, for paleoecological, paleoceanic studies as well. But currently, we want to know what is found in the country recently in terms of these organisms and in terms of plankton and whatnot. Next slide. Now, just to share with you the study methodology of these organisms is again, they're part of plankton. So we usually use phytoplankton networks. These projects, these meshes, these projects are. We're starting with one project in Isla del Coco with samples that had been extracted back in 2008 and all through 2012. These are projects for the radiolarians that I have at SAMIC myself, and now with the Biomar project, we're working with these plankton samples mainly for the taxonomic identification to see what's what's out there, and also the genetic part too to see if we're able to work with this as well. This is another matter.
And we also have sediments, which is what we use the most. As Jorge mentioned, he has also participated in several expeditions, as many biologists, and they bring us samplings, and we obtain that in the most outer layer, which is the most recent one, and that's where we look into the different microorganisms of foraminifera and radiolaries. And here, taxonomical, and in this the first portion of the square, we have some radiolaries from the tropic and subtropic, and we identify what we have in our country, and to determine the ocean, the oceanographic and the ecological. They can be they can go up down to 3,000 meters of depth. So that also helps us to have some indicators of electricity, oceanographic information, depending on the temperature as ecological indicators. And then we also have the location of the study sites that we've been working on. We have all of these samplings all the way from the Osa Peninsula, Isla del Caño, Coco Island, submarine mounts, depths from 40 meters to 40,000 meters, and we have the submarine pampa, which is what shows here along with the sediments. And so I will show you what we've been working on. This is part of the thesis for the license of Alexis Rodriguez. And what happened was the taxonomy of radiolaries from 15,000 years till current, and also the submarine mounts. This is something that has already been accepted. It will be published in the Tropical Biodiversity magazine. And we also have a glossary with a descriptive morphology, kind of what you can see here. So you can see images of microscopic images. So you can use that whenever you have plankton studies or something related. The species don't really vary from 10,000 years till now. Yes, our work is also with recent studies, but it's the same. And we also have the taxonomy of radiolaries. We have approximately 42 species. And then Nasalarios, those are the ones that look like little cones. And then Colodaria, about 6 species. And then we have um, this other study that we've been uh, moving forward with the geology study. She's studying specifically Moniferous, and it's quite detailed. There's a nucleus, 15 centimeters, and each centimeter is studied specifically, and all of the species come up there. And I will show you the superficial species, which are of the biological interest. And and then we have this image here where we can see the location, and this is regarding bentonic forms, and they provide some changes in the environment of low oxygen in the sediments. And when we see the plantonic forms, we have approximately 20 species of those, and they are also indicative of variations in marine suggestions, changes in nutrients, and so forth. And in the submarine pampa, this place here is in El Dorado, where the octopuses were, and where this expedition was taken. We haven't found any of these plankton species in this specific area. We have only found forms that don't even form a skeleton with calcium, but with is in the extract. So they just obtain some of the nutrients that surrounds them and build their own shells. And we could continue to work in different sampling, but this is superficially what we have. And this is around 3,500 meters of depth. And then we have the necessities for these studies. The students with interest, especially in biology, in geology, we have people that are interested, but we do still need people that are interested and to work the plankton as well. We have the modern team from the plankton network. We need a modern equipment, and then genetic studies. I'm not at all specialist, but we are working with the specialized biologists. We need studies in different shallow areas. We have some, but we need more. Then we also have the radiometric data, because we love to know the age of everything. And then the revision of sampling of existing projects. We need to know with how many thousands of years we're working with, because you might have taken a sampling, but we don't know the sedimentation rate or how long, how much time there is in that 1 centimeter or 5 centimeters. So that's very important. And then the revision of sampling of existing projects. I know in biology there's a lot of people that obtain sampling of plankton from different areas, and I feel that would be great to have more assistants, more students, more projects where we can revise the sampling and we can amplify the diversity, the marine diversity of this group. Next. Okay, well, thank you so much. This is a radiolary. It's very pretty. Thank you. We will open a section of questions and answers. Does anyone have a question? Okay, well, hello, good morning, excellent, everything, and thank you so much for all the work you all do. And thank you for all the information on the collections that I believe is super interesting. And in that, on that, I would like to consult with the collections. There's a lot of challenges in curations and to obtain the right space for storage, temperatures, and so forth. And I'd like to know what challenges you faced in that area and maybe challenges you've overcome. So I would like to know a little bit about that experience regarding those challenges. Yes, it's very important to maintain stable conditions in museums for temperature and light. There are samplings that lose color. We have pest control because we are at war with ants and other types of ants that in certain areas, certain times during the year, they come and try to obtain the samplings or come near. There have been some bacterial layers that we've had in the alcohol sampling. So with those examples, we know we need to change the alcohol in the moist collection, and for the dry ones, we do the pest control. So it is a big challenge to obtain and maintain the correct environment for the collection for the samples. And so yes, we are on top of all of that. There is someone who handles the collection and cares for them, and they have support for assistants, volunteers who come every year and help us to change the alcohol. You can imagine all of the jars that we have there. And you're all invited whenever somebody wants to come by. We can definitely schedule a visit. We don't have an exhibition, but we can definitely schedule visits just to see what we have there and maybe help out with something. But yes, it is definitely a challenge. We need the appropriate conditions, and it's quite costly as well. And yes, and also for the herbery, it's the exact same thing. We have some moist collection. We have alcohol and jars as well, and the funding is something that's important and that the university does, and really the whole Costa Rica as a whole provides these funds, and so because this is ours, and in the specific case of the herbery, for the dry collection, there's a specific paper we need. And then bit by bit, we've been training different students for these different methodologies and techniques. So we need to invest time. Every day, there's something that we need to do. So yes, it's an investment and the collection of biodiversity in our country. So yeah. Some years ago, I was very much involved with the museum, and it was impressive. Everything we needed, and the budget as well. And one of the things that I think it has improved is to understand, and I think we've all understood that the authorities maybe didn't understand why they needed to invest thousands of colones in a whole bunch of jars in a basement. So that's important, and it still is important, and I'm sure Monica constantly needs to lobby for those budgets, but that's something that's needed, and we have specimens there from the 19th century, even some before the National Museum. So some birds were there from the 19th century. So samples that show us that that bird was in that place in those dates. Yolanda? Yes, well, I also wanted to add um, that in the last years, the collections have also been affected by the climate and and when what we understand, which is the export and import of different specimens that we lend. That's been complicated, it's not clear, and currently we have some connections that belong to the country, and we need to bring that back. And the process to do so is quite heavy, and the taxonomic value of those collections is also an important factor. And yes, I'm sorry to include myself, but I have an idea that I've had for some years. It's been some years where I feel like the country should have a museum of natural history, and where we could see the evolution of different specimens and bring all those collections together that are kind of just dispersed through different museums, small museums throughout the country. And I feel like we should have like that policy in the country to observe, to know, and have a space where people can come and visit and show that as a scientific campaign. That's something I've always dreamt with. There are many people. We're kind of maybe dispersed, but maybe the China embassy will help out. Maybe that's a project, an alliance between different universities, and sure, there's a cost for everyone, but it would have a great projection for society. Thank you. Anything else? No? Okay, I would just like to say, I would like to close this panel with a question for everyone and for anyone who would like to respond. How can these this data that we've presented contribute to the formation of public policy to address that? Yolanda mentioned something which is very important, but what else do we need besides what we already have to contribute to the marine diversity of our country? Well, something important is the campaign, divulsion, and try to generate that this knowledge doesn't just stay in the academic genre, but also give that to the people. And I believe education is key there, maybe some sort of policy that takes this knowledge to the knowledge of the country, schools, high school, and then that strengthens the collections in the museums, in the National Museum or in the UCR. The subject of biodiversity is important because we need to see how the group, there's a lot of subgroups, not everyone needs to know everything, but as a general population, if we understand biodiversity, we're a better population than those who might not know anything. So I believe that this campaign and really just divulge this information is important. Some time ago, I wore another hat. I played another role, and I would like to go back to that and just understand who needs to be completely convinced is the president and the ministry for tax authorities. So how do we convince them? Well, we would hope that the fact that they're students and colleagues that maybe rise to obtain these positions where they make decisions. And then us as scientists, we need to get involved more in politics. Sometimes we don't want to have any type of relationship with that, with politics. With me, sometimes I think, that's just going to be a headache, it's going to be a problem. But if we don't do something, the system will continue the way it always has. So then it's the pressure from society, and for there to be a public policy, really, it's just two people, the president and the tax ministry. And then the next is also, for example, the Legislative Assembly. There was a policy that said, no, your deputy. And they would go and speak with this deputy and let them know about their project, but there's really not a lot of people. Maybe they're not convinced, maybe they don't know, and then they won't really do anything about it. So that's really sad. That's my opinion on that. Okay, great. Very interesting. Any other question? All right. Well, I'd like to thank you for your presence in this panel and all the presenters and guests as well. Thank you. Good morning
Vamos a dar inicio
to everyone.
al
We
último
will
panel
begin the last
que
panel for
por
the
lo menos
day,
la mesa
at least for
que lo
the
organizó
organizing
la Universidad de Costa Rica en el marco de
committee that the
este
University of Costa
Congreso
Rica
de Integración de
placed within
Saberes
this integration
para un océano
of
sostenible.
knowledge for a sustainable
Antes de
ocean.
presentarles a los
Before
panelistas,
presenting the panelists,
solamente decirles
just
rápidamente,
to let you know
mi nombre es
quickly that
Álvaro
my name
Morales
is
Ramírez,
Alvaro Morales Ramirez.
soy el director del
I
CIMAC
am
de la
the
Universidad
director
de
of the
Costa Rica y voy a
CIMAC
tener el honor
for
de
the
moderar
University of Costa Rica, and I will be moderating this panel,
que es sobre un tema
which is
muy
about a
pertinente
subject that's very pertinent
sobre la restauración
regarding
ecológica
the ecological
marinocostera
restoration
como
of the marine
herramienta
and the marine
de
coast as a
sostenibilidad
tool
ambiental.
for sustainability,
Y para eso
environmental
hemos
sustainability,
invitado
and
a
for
4
that we have
personas
invited
que
4 people
han desarrollado
that have
una
developed
actividad importante
an important
en el marco de
activity
los
within
procesos
the
de
different
restauración, una
processes
vasta experiencia.
with a
En
large
primer lugar voy a
experience.
presentarles a
So,
la
for
doctora
starters, I will
Jimena
present
Villarreal.
Dr.
Ella va a ser la
Jompera
primera
Samper.
expositora.
She will be the first
Ella
presenter.
tiene un bachillerato en biología
She has a
en la
license
Universidad de Costa
in
Rica,
biology.
una maestría con honores en manejo costero tropical
She also
de la
has a
Universidad de Newcastle,
management of
en
coast
Reino
from
Unido, y un
Newcastle
doctorado
from the
de la
UK
Universidad de
and a
Queensland
doctorate
en
from the
Australia.
University of
Es
Queensland in Australia.
investigadora del CIMAR,
She is an investigator of CIMAR.
Coordina la Comisión
She coordinates
Institucional de
the
Biodiversidad de la
Institutional
Universidad de Costa
Committee
Rica
of
y el
Biodiversity in Costa Rica and
interuniversitario de
the
CONARE.
inter-university
referente a la
team
gestión de la
from CONARI
biodiversidad.
regarding the
Coordinadora y fundadora de la
biodiversity.
Red
She's a
Tropical
coordinator of the
de
Tropical Network
Restauración,
for Restoration,
Red Regional para la Restauración de Pastos
regional
Marinos y
for
Humedales.
marine pastures,
Es miembro de la Sociedad para la Restauración
and
Ecológica,
she's also
miembro del Grupo de Trabajo Técnico de
and also
Carbono Azul,
mangroves. She
de la
also
Liga de
is
los
part
Océanos, de las Naciones
of
Unidas,
another
y
team
miembro del
from the
Comité
United
Ejecutivo de la
Nations
Asociación
for
Mundial de
marine
Pastos
conservation,
Marinos.
and
Además, miembro y líder
she's also a member and a
regional
leader
del Grupo
of
Especialistas de Especies
the
de
regional
Pastos Marinos
team
para la
for
UICN.
in the UECN.
La segunda persona que
And the second
va a
person
intervenir
who will
es el
present is
Máster Lenín Corrales.
Master Lenin Corrales.
Lenín tiene un
Lenin
bachillerato
has
en
a
ciencias biológicas
doctorate
y
in
una licenciatura en
biological
biotecnología de la
science,
Universidad Nacional y una maestría en
and
tecnología
a
de
master's
información.
in technology
Ha
for
realizado
information.
estudios de
She
especialización en
has
manejo y
conducted
evaluación de recursos naturales
studies of
y impacto
investigation
ambiental en varias
in
instituciones
handling natural
internacionales.
resources and
Su
also
experiencia
in
laboral
several
se centra en América
international
Latina y el
universities.
Caribe,
Her
con un
experiences in
enfoque en
Latin America
Centroamérica.
and the Caribbean with a focus in Central
Abordando
America,
biología de la conservación
boarding
y
with an
restauración
approach
de
for
ecosistemas. Tiene
ecosystems.
39 años de
She
experiencia en
has
biología de
39
la
of
conservación
experience
y 24
of biology
en
and
cambio
conservation
climático y ha sido
and 24
investigador en
in
biodiversidad
climate
en
change
Costa
and has
Rica
also
para
been an
el Estado de
investigator
la Nación durante 15 años.
for the
Actualmente es
state
asesor
of the
senior
nation for
en la Unidad
over
de
15
Acción
years
Climática del CATI.
and works in the CATIE.
La tercera persona
The
que
third
va a
person
intervenir
that will
es
intervene,
María José Solano González.
Maria Jose Solano
Ella es
Gonzalez.
bachiller en biología
She has a
y forma
doctorate
parte- y
in
es
biology
parte
and
del
is part
Laboratorio de
of
Ecología y
the
Conservación en
laboratory
Arrecifes
in
Neotropicales del CIMAR. Actualmente es
CIOMAR,
estudiante de licenciatura en zoología y
in
estudiante de la maestría
reefs, and
académica en
she's
gestión
also
integrada de áreas costeras
has a
tropicales.
Master of
Ambas
Tropical
en la
Regions,
Universidad de Costa Rica.
both from the
Y desde
University
hace
of
3
Costa
años
Rica,
colabora en un proyecto de
and over 3
restauración
years ago
coralina
she
localizado en Playa Culebra,
cooperates with a
lo que
project
le ha
in
ayudado a
Guanacaste,
sentar las bases
which has helped
de su
her
proyecto de tesis
with her
en la
thesis
maestría
in the
con
master's
colaboración directa
with a
con la
direct
comunidad de Ocotal y
cooperation
Guanacaste.
from the Oacutal community in
por último,
Guanacaste. And
Tenemos la
lastly, we have
intervención
the intervention
de la máster
of
Jacqueline Rivera,
Master
que ha hecho un
Jacqueline
área del
Rivera.
MINAE
She
desde
is
hace 18
an
años.
employee of the MINAI for over 18 years.
Es asesora técnica,
She is a
fue
technical
asesora
advisor,
técnica,
or
bueno,
she
es que
was.
ya no existe, ¿verdad?
Well, it's just that
Ese
that
ministerio.
institute no
Pero
longer
mientras
exists.
existió,
But while it
ella
existed,
fue
she
asesora
was
técnica
a
del
technical consultant
Ministerio de Aguas y
of
Mares,
the
con experiencia en la
Vice
generación
Ministry
de
of
política
Water and
pública, como
Ocean,
la política nacional
and
del mar
how
y la
the
política nacional
policy
minerales, entre otras.
of
Actualmente labora en el Sistema Nacional
different
de Áreas de
policies of
Conservación
the
como
ocean and
coordinadora nacional
also
en el Programa
for
Nacional de
the
Humedales y punto
wetlands.
focal ante la Convención de Ramsar. Es
And
ingeniera
she's
forestal, graduada en
a
Instituto
forest
Tecnológico,
engineer.
licenciada en Manejo de
She's
Recursos
a
Naturales de la
license
UNED
in
y máster
natural
en Gestión y
resource
Auditorías
and master
Ambientales
in
con
different
énfasis en Cambio
auditory
Climático de la
uh,
Universidad
in
Internacional Iberoamericana
different
en Puerto Rico.
environment studies in Puerto Rico.
Cada
And
una de ellas
each of these presenters
las
will have,
personas
each of these
tendrá la oportunidad de compartir
people will
con
have
nosotros por 10 minutos
10 minutes,
y al final
and then toward
vamos
the
a
end,
tener la
we will
oportunidad
have
de plantear nuestras
uh,
preguntas o
a, an
comentarios
area for
al
questions.
respecto. Entonces, Jimena.
And so, Jimena. Thank you for that presentation. As those who are also with us virtually, as presented, my name is Jimena. I will speak about the restoration for the environments of blue carbon in Costa Rica as a tool, as a source for sustainability. Planet Earth is 92% ocean, as well as our country, and we have a large portion of this. However, there has been a large degradation and loss of these environments, specifically by anthropological effects, some local, some are regional, but there has been a deterioration of these. And so we need to think not only of conservation, but go further and think on how can we recover these ecosystems for these different environments. This is really well established as to the decade of restoration for ecosystems for the UN, and we have different policies focused in the restoration for the environments for lands and also marine. And our focus of restoration of the restoration is regarding coral reefs, the mangrove swamps, forests. There is an analysis that we will present of the restoration and the plans that we have in our country to see which has been more successful, which one hasn't, maybe. And then we have the other ecosystem that we're dealing with is marine grasslands. So the swamps and the grasslands are blue carbon, which means that they obtain the CO2 and they leave that in their sediments, as well as carbon from other sources, and they store it there not for decades but hundreds to thousands of years. So this has really been great for the climate change, and that has been a great advantage to this. For those who aren't familiarized with marine pastures, they are vascular plants. They have flowers. They have many characteristics. We found them in both coasts, different species, and they have different strategies for life. And in the Pacific and in the Caribbean, we've seen the degradation and the disappearing of these species, and we know this due to the environmental monitoring that we've had in these environments. So we see that we've been monitoring them since 2010. In the North Pacific, we had the disappearance of a pasture around 1990s due to a storm. That's kind of what the landscape was before. That's a photograph of Dr. Leon Cortez. And because that disappeared in certain times, we've tried to help them recover naturally, but to the date, we still have not had that recovered. It's more of a sand surface, and we can find some seaweed. And so then we started a natural recovery project, and what we started was try to understand why it wasn't recovering naturally in this place, because nearby we do have some pastures that have been developed. So we look into the quality of water, excessive herbivores, to see if there are seeds, or else maybe place some seeds there that can germinate naturally, and then also try to identify transplants for different areas that may be ideal for pastures, and so try and transplant that and help nature recover itself. Next. The other things we've done is try to understand the species that we have, which is the um, treble pasture, and that is in a red alert. But we are looking into different studies for different pastures in our area, and also with Belize and Brazil, to try to understand how this species disperses itself, because we only find it in one specific place, and it's very fragmented, so it's hard to lead a project with that. So we try to understand different techniques for transplant and to cultivate that to help the different um, way that it works, temperature, salinity, production of light, and all of this in some aquariums. In the Caribbean, we monitor some marine pastures. Near the end of the project of the CARICOMP, which we monitor long term, and we can see it on the right where the pastures were monitored, and some disappeared and some they remained. In our country where we monitored, we've seen a degradation and a disappearance of the pasture that we had. So before, it was the the field had large leaves, and it went changing to thinner and smaller leaves, some pioneer species, and then currently it disappeared. Thanks to a thesis for a student in Costa Rica, Jairo Moya, we, in the restoration site, but also in a spatial area of Cahuita, from 2019 to 2021, there is a reduction of the marine pastures of almost 50%. And that's because there has been an excess of herbivores, specifically the green turtles that have been eating the pastures. So one of the studies that we did initially was place sort of a cage to protect from the excessive herbivores. And you could see that within a month, there was so much more pasture, greater leaves, stronger, and the dominant species was there. And then outside of the cage, it was it was a much lower. And then we have the remains of pastures, and that's a strategy that we can try we are trying to coordinate with the local committee and with the SINAC so that it's September, October, and begin this process within the National Park of Cahuita. We also want to let you know that you can join to our distribution network, the Tropical Restoration Network in the Caribbean, in the Pacific Caribbean, Oriental Pacific Caribbean. We have a restoration of mangrove swamps, marine pastures. We have workshops, webinars, courses. Next month, we will have a course of restoration for these mangroves. So with that, I would like to thank all of you.
Muchas gracias.
Thank you,
Jimena. Y le damos la
Jimena.
palabra
And now,
a don Lenín.
Don Lenin. Thank you. Really, I was given a task to let you know a little bit about the state and the challenges of the restoration of the mangroves that we have in our country and how the reconstruction, historic reconstruction in sight to see how much potential we have and why these mangroves have disappeared. Since May 22, we have another reason as to why we need to work in the restoration of these mangroves. The UCN placed it in a red list for ecosystems where the mangroves of Costa Rica, of the Pacific, and the Caribbean are considered vulnerable after the evaluation, after the assessment that we've had on a global scale. In the last job we had with the ecosystem of these mangroves, we established a map that was set up with a satellite of a high resolution in the country, and that has a total of 52,800 hectares in the country. And something important to point out is that this is the first time that there's been a map of the ecosystem. So it includes not only the coverage of the forest per se, but also all the other elements that have these mangroves, beaches, alpine areas. And historically, what we've been doing is a map of these swamps where we can only just see the forest, but not just the composition, not the entire composition. So due to that estimate, we were able to set up a difference of different types of mangroves, high, low, and other important elements, different use that different companies that deal with shrimp or salinity work with, and then also another area of black lining that's set up in these mangroves, channels, lagoons. And what we've handled so far, we've been handling data. There was a recollection of different air photographs, one of the first ones that we have of the country. And right now we have an estimate that there was 67,000 hectares of mangroves in the 60s. If you subtract that to what we have nowadays, we would think, okay, so we have a potential of restoration of nearly 14,000 hectares. However, that in the analysis we've seen as to why this has disappeared, why they have diminished these mangroves, and why they continue to disappear. And really, the mangroves in Costa Rica have different factors as to why they have been degrading. Before, we thought it was the companies that worked with shrimps or such, but the expansion of rice, of sugarcane, of the African palm, that was provided on several portions of the country, and lately the expansion of the coast hotels that has also affected.
Taking away chunks of the mangrove, and we also find in some areas of the country that we're losing mangrove because of the marine weather, meaning an increase in the level of the waves, which is eroding away the frontal areas of mangroves, mainly in the southern part of the country. This is seen when multi-temporal analyses are done. Next slide. Why did we begin saying that there aren't really 14,000 hectares of potential restoration available? And it's because we have mangrove areas where the restoration process is completely irreversible. An example of these would be the mangrove in 1960 in Punta Arenas near Chacarita, for those of you who are familiar with Costa Rica's geography. And in 2022, all of these little yellow dots are roofs, meaning that the city of Punta Arenas is expanding. Um, just like this, we have different such as Quepos, for example, that expanded over mangrove areas. We also have roads in the Central Pacific, for example, when the coastal highway was built, and mangrove areas had to disappear for this. So we don't really have 14,000 potential hectares if you see really gross numbers, and we're truly unaware because we really have to tune down what truly could be restored. Next slide. Here, I would like to start by reflecting a little on the challenges that we have for mangrove restoration. In Costa Rica, in the latest years, we've had a lot of empowerment and boosting, and many organizations are working on mangrove restoration. But when one collects these experiences, applies certain criteria to it, then one starts realizing that we still need to start organizing the whole topic of restoration a little more. So one of the things that we should do is to truly establish what is the restoration process, because what we've seen is that there are different methods, and one of the processes that have been used is precisely restoration based on plantation and scientific literature and based on studies of many years. And so that it doesn't happen as it has happened in other countries of the world, the studies truly show that the long-term success of this type of restoration only goes in between 1.52% to 18% of survivability for the future. And there are also publishings of the um, losses of millions of dollars that have been invested in restoration, basically because of the fact that there is not an organization behind these processes. And what is recommended when mangrove plantations need to be done is that we need to have that extreme step that we should be doing. The preliminary estimations of restoration is incomplete data because in the database that the wetlands national committee is doing, there's still some data missing, but there's around 458 reported hectares with restoration actions and about 50 restoration sites. On the right, we can see the size ranges of these intervention sites, where 65% or more than 65% are truly sites that go between 0.6 hectares to 5 hectares in size, right? And There are only two experiences. One in which there is an area of 129 hectares that which used to be a sugarcane plantation, and that represents the largest site of restoration in the country. The rest are just small units on their own. Um, Next slide. There is another important aspect. What do we understand as ecological restoration and the recuperational um, management of the mangrove? This graph that you see on screen is precisely of a research that the student is who did it is also finalizing her thesis. She is from Honduras, and she started her work precisely by interpreting the succession process of mangroves in the Pacific, and what she has found by studying the story of when the process truly began. What she has seen is that many sites that in nowadays are accounted as restoration sites are truly sites that have been under passive restoration for 12, 14 years, and that simply during a specific year we opened a wall, more water flowed in, and then it went into the restoration statistic. But in terms of these restoration sites have more than a decade of being restoration. So it's important to define when and what type of project frustration have to be considered, what sort of criteria should be defined in order to consider these. And something very important that we're noticing is how much accompaniment has to be given to a restoration process, because what we are noticing is that restoration processes um, require investment, and after that it seems as if the process is finalized and then nature has to take care of finalizing the task. But truth be told, what we are seeing is this is an example of a recent event where the restoration process that was taking place in an old sugarcane field was simply set on fire. So the whole area of the restoration process was lost. We have another example where basically people simply cut down all of the mangrove that had been in restoration for 3 or 4 years. So this issues out an alert to truly establish deadlines and demands of for how long a restoration process has to be accompanied, because it's simply unbearable that we just simply abandon it, and there has to be aspects of maintenance and of continuous vigilance considered as well. Next slide, please. Another thing that we have assessed are the costs of restoration, and these are very diverse. For example, we have costs that go from $19,000 a hectare all to $179 a hectare. So there is definitely diversity here. We truly have to work in the estimation of costs, because we can't sell restoration with values of $19,000 per hectare, because it's impossible. It's impossible for budgets. And what we're seeing, and this is a little bit related also with the shape in which restoration projects have been installed lately, because these are based on projects. So by being based on projects, the experience tells us that values get inflated, and this can also be a bad sign for future financing, because something that we have discovered is that when restoration projects and please colleagues at NGOs forgive me, but when restoration projects are handled by NGOs, these become extremely costly. And I believe that we have to work on this in order to not transmit these messages that certain groups of society are too expensive when doing restoration. So there's this whole question of how much restoration truly costs. We have done fine work in costs, and basically in old cane plantations, the cost is around 1,100, $1,200 per hectare, and on salt mines and shrimp companies, similar under very strict methodologies for costs. Um, Almost wrapping up here, I have, and in order to conclude, I believe that we really have a necessity of establishing mangrove restoration principles that are based in, for example, based in first correcting our hydrology. We can't just simply plant or knock down a wall. We need to research on the root causes as to why. With research that is being done in my particular school where I work at, we're working on the chemical composition of soils and waters based on or the origin of the disturbance. We have to correct those adverse conditions. We have to use reference sites as well. We have to understand the topography, and something very important that we are abandoning is the principle of building um, tidal creeks, which is the opportunity that fauna is going to find, so that when the tide flows in and then flows back, they can travel on these, let's say, highways or roads. Because what we're seeing is that since there are no channels within the restoration projects, then the tide goes away, and small fish don't have enough time to exit along with the tide. So this is a very important principle. The principle of assessing restoration costs and adequately documenting the projects. We have a very big deficiency in the documentation of projects. And lastly, a last principle is that follow-up has to be provided in the short and midterm. Okay, so this was a very, very brief. I only had 10 minutes to share with you the story of what is going on in the country in terms of restoration. Thank you so much, Lyndon. Maria Jose? Yeah, that one. Can you all hear me? Perfect. As Mr. Alvaro was mentioning, I am a master's degree student, and part of what I'm going to be presenting here are some of the preliminary results that we have from my thesis, in which we're working collectively along with the community in Ocotal, Guanacaste, and we're doing a citizen science process. We're basically teaching the community of Ocotal and Playas del Coco community as well to basically what are the reef restoration techniques and also how to monitor these ones. Part of what I wanted to mention from the get-go is that this work would be impossible without the collaboration of the dozens of people involved in this project already. Everyone from their different trenches are working so that this project truly works. Um, Firstly, it's important to understand what citizen science is. We can visualize this as research processes in which individuals who perhaps didn't attend a university or obtain the science degree get involved in research processes. Each author defines as what they understand as citizen sciences, but we can conceptualize it um, just like that. We have spoken about the benefits of citizen science and for research, and I particularly like the accommodated into these two criteria. First, because it's a participative process in which in many occasions it serves as a platform for environmental education. It also lets us capture the social dynamics that sometimes from the academic perspective get excluded from research processes, and it's also a way to give communities access to science, and in this case coastal communities. And on the other side, which is also very attractive for us, is that it generates a lot of scientific data as well. Of course, there are studies that have validated the fact that data collected by citizen scientists can be of as much quality as those of academic scientists, let's say. And these becomes more tangible when there is a training and accompaniment from the academic side as well. Also, the projects that have the projects that have citizen science involved increases the amount of time that we invest sampling in much more broader areas, and this is also related to another point, which is reducing research costs, because we have noticed that there's a reduction in cost, and many here know that financing is one of the biggest problems that we face when doing research. This picture is of a coral reef. in Culebra Bay, these coral reefs are the ecosystems on which citizen science projects have been done, not only for restoration but also for monitoring for invasive species as well. These pictures were taken with a month's time difference. These were taken um, by the middle of last year, and these talk about the current situation that we have with coral reefs. NOAA recently declared the fourth largest uh, bleaching effect worldwide, so this could also reflect the effects of the loss of these ecosystemic systems. Now, moving over to Ocotal, Guanacaste. Ocotal is located in the North Pacific region of Costa Rica and has a very shared story with many other coral reefs of the region where multiple stress factors, global and local factors, acted so that there was a very strong degradation of these ecosystems. Amongst these stress factors are poor practices of coastal development, kelps on an increase in marine temperature, which took 44% um, diminishment for values in 2010 of 1 to 4% coverage. This is why in 2019 there was a restoration initiative started in which we are putting fragments from other reefs in these structures or structures that are similar to these ones in order to provide maintenance to them and have an active restoration process in 2023, which is when we started working with this project of citizen science. We gave this more social component to this whole process. The Alcatel project, we can frame it in what is known as a collaborative project in which there is participation from these citizen scientists well beyond just sampling, but with the biggest investment from their end. This is a little bit of the story that we have so far. The project officially started in February 2023 with a community workshop, but since April, all through May, since April of last year, all through May of this year, we have been holding events, and these events have not been only uh, plantings or maintenance, but also trainings, monitorings, and community activities as well. In the beginning, we started with 15 structures only, and this number was increased in August. By August, we had 45 in Ocotal only, and these came from university funds and also private donations from local entities mainly. By September, one of our key partners was Garnier and Garnier developer, which provided very strong support in terms of financial support, and it allowed us to reach the 113 nurseries that we have, reef nurseries that we have, and these represent approximately 2,260 live fragments that are currently growing, and it also allowed us to open, let's say, another restoration point in Punta Cacique. Currently, there are more than 30 local divers who are involved in this process, and this is a process that is continually growing. Next slide. We have faced challenges. I believe that everyone who is here present and is related to preservation know that there are challenges. One of these is that since we're teaching the community how to do monitoring, it's something that is transitional. It's not something that is thought in a day. We have to do trainings every time a monitoring is going to happen. But with time, we've seen that how even for volunteers, it becomes more simple and more simple to learn more and more things. There are difficulties that are specifically of working in the ocean, such as scientific diving, just diving in general, and also safety, because of course we want this to be a safe activity for those involved. And another thing is that by November of last year, we reached the maximum point of whitening, let's say, where 83% of our colonies had whitened. But with good news, that this whitened, that this whitening didn't represent mortality. There was mortality present definitely from fragments of two specific sites, but the majority survived and are currently growing. And I'd like to highlight basically that it has been proposed that citizens science has a role within conservation, and these are six different criteria that authors indicate that don't have to be seen as isolated criteria, but as a dynamic activities that can include many of these. Part of what we look for in the Ocotal project is for it to be functional, for it to be a platform of environmental education in which the people who are coming closer to this project learn about coral reefs, but also for they to become citizens who think really well about decisions in their lives and the way that they interact with ecosystems, and for it to also serve as a platform for stakeholders that are involved to interact um, in a fluent manner, let's say, and for this in the long run to help us to manage these type of ecosystems. This was part of what I was previously mentioning. This is a picture from November of last year in which 100% of the fragments were pale or whitened. And next slide. And this is May of this year. There was a recovery. There were fragments that unfortunately did die, but the corals that we currently have are growing now. In this graph, we're seeing the updated results all through December of last year. The yellow section represents whitening, but from December and on, we can see recovery represented. And for the following, you're gonna have to trust my word, but all the fragments that we currently have some good coloring of their own. And now to wrap up, I would like to highlight one of the pieces of data that actually comes from one of the surveys that I'm currently doing. 94% of individuals who were surveyed to this date consider that the restoration of coral reefs in the North Pacific area is tremendously important. Sorry, I got no mic. And this is also a call for action that this citizen science tool can be used to restore these type of ecosystems, and not only reefs but other ecosystems as well. And that's it.
Muchas gracias, María
Thank you
José.
so very much, Maria Jose.
Tenemos a Jackie.
And lastly, we have Jacqueline.
Muy buenos días a todos.
Good morning to
Bueno,
everyone.
del Sistema Nacional de
From
Áreas
the
de
National
Conservación, es un
System
gusto
of
poder
Conservation Areas,
compartir
it's
aquí
really
en
a pleasure
este
to be able
espacio
to
que son
share the space with all of you.
Bueno, vamos a ver, seguro
Okay.
porque se acerca la hora del almuerzo.
Okay,
debe
it
ser.
might be because lunch is
Como les decía,
almost upon
desde
us.
el SINAC,
As I was
muy
saying, from
contentos,
SINAC, we
de
are
compartir
very
en este
pleased
espacio
to
nuestra
share
institución.
our institution with everyone.
¿Todo bien?
¿Mm?
¿Estás bien?
Mhm.
Entonces te cambio en sí.
Gracias.
¿Mm?
Ahora
Sí.
sí.
Okay,
Retomamos
there we go.
por
Picking,
tercera vez.
picking back up for the third time.
Como les decía, desde
As
SINAC
I
muy
was
contentos de
mentioning,
compartir con
we're
ustedes
all
en
very
este
pleased
espacio.
at SINAC to share the space with
Muchísimo
all of
trabajo
you.
conjunto con
There's a lot
la
of
academia, la
collective
Universidad
work with
Nacional,
National
con
University,
el
with
CIMAR,
academic
que siempre
sectors,
han
with
sido
CIMAR, who
amigos
have
cercanos,
always
¿verdad? De
been very
trabajo, de
close
propuestas
work colleagues
de
for
generación de
proposals
ciencia para mejorar la
generate
toma de
science in
decisiones.
order to improve decision
Otra
making.
cosa muy cortita también que no
Another
puedo
short
iniciar la
thing that I
presentación
can
sin decir
definitely
es
not start
que,
my
bueno, me
presentation without
alegra ver
mentioning is
a
that I
muchos
am
compañeros del
really
SINAC,
pleased
pero
to see
también
many colleagues
a
from CNAC,
colegas que
but
trabajan en
also
conjunto este tema de
colleagues who
manglares. Yo
work
hoy
collectively with
simplemente soy
mangroves.
una vocera.
Today, I'm just a
El
spokeswoman.
avance que nos presentó Lenín,
The
mucho de lo que
advances
se ha
that Mr. Lenin
trabajado
presented,
con el CIMAR, ¿verdad?
much
Con
of what has
Jimena,
been worked
hasta estos
at CIMAR
proyectos
along with
en
Jimena, with
Ocotal,
these
realmente no
projects in
serían posibles
Ocotal, they would
sin la
not be
coordinación y
possible
sin el trabajo
without
conjunto,
the
¿verdad?
coordination
Entonces,
and
me
collective
alegra
work.
mucho ver tantas
So
caras conocidas
again,
y
I am
también
very pleased
otras
to see so
nuevas
many
y hacer la
known
invitación a
faces
que
and
este
many
tema
new
de
ones as
restauración de
well. And to
ecosistemas
those I'd like to
costeros es
invite, that
demasiado grande,
this whole
tenemos
topic
muchos
of coastal
retos y
restoration is
todos son
very big.
bienvenidos
There are
a
many
trabajar, ¿verdad? En
challenges,
este gran
so you
equipo
are
nacional.
all welcome to
El
work
día
in
de
this
hoy
big
quiero
national team.
compartirles un poco,
Today, I'd like to
una síntesis
share
realmente,
just
porque
a
los 10 minutos no
synthesis, I
alcanzan
would say,
tanto,
because the
sobre la
10 minutes are not
rehabilitación
enough
de esos
about the
ecosistemas de reservorios de
rehabilitation
carbono
of
azul. Ya
these
lo vimos con
ecosystems, blue carbon
Jimena, ¿verdad? Son
reservoirs. We
manglares,
saw with
pastos
Jimena,
marinos,
these are
marismas
mangroves,
y hasta turberas
seagrasses,
costeras. Y por supuesto que
seamounts,
por ahí los arrecifes de
and of
coral, aunque
course,
no
coral
fijan,
reefs.
pero sí tienen
Despite
una
not
interrelación demasiado
fixating,
importante
they have a
con
very
los ecosistemas
important
costeros. Entonces,
interrelation with
cómo todo este
coastal
engranaje
ecosystems.
se
So
alinea con la
how
política
all of these
pública a nivel
aligns
nacional.
with
Siguiente,
national
por favor.
policy at a national level. Next, please.
Bueno, como dijeron en la presentación, también tengo
As
otro sombrero,
mentioned in
además
the
en el
presentation, I
SINAC,
have
¿verdad? Que
another
es
hat that I
hacer
wear at SINAC, which
este
is
enlace con la
about
Secretaría de Ramsar, que es la
linking with the
Convención
Ramsar
de los, a
Secretariat,
nivel
which is the
internacional de los
International
humedales.
Convention of
Hace
Wetlands.
pocos años, relativamente
A few
en el
years back
2018,
in
la Conferencia de las
2018,
Partes
in
aprueba
the
una resolución
Conference of Parts, there is
específica
a
para
specific
promover la
resolution approved in
conservación y
order
la
to
generación
promote
de información
conservation and
relacionado
generation
a
of
los
information
ecosistemas
related
reservorios de
to
carbono azul. Esto
blue
es sumamente
carbon reservoir
importante, puede ser
ecosystems.
solo
This
un
is
papel,
very
¿verdad? Un
important. It
documento,
might
pero
just be a piece of
realmente
paper, a
pone
document, but it
en la
really
mesa
puts on
política de
the
muchos
table
países el
the
compromiso
policies
de
for many
comenzar a
countries and the
movilizar o
commitment to start
mejorar el
mobilizing
trabajo que se
or
esté haciendo
improving
dependiendo
the
de la
work
realidad de
that
cada
is being
país.
done depending
En
on
el
the
caso de
reality of each
Costa
country.
Rica, el
In
trabajo de
the case of
manglares
Costa
no es
Rica, the
nuevo,
work on
¿verdad?
mangroves is
Pero
nothing
sí
new.
necesitaba, siempre
However,
necesita ser
it
fortalecido
always needs to
y
be
muchos hemos
continually
sido
strengthened.
testigos y
And many of
también
us have
actores de la
been witnesses
movilización de
and
este trabajo
stakeholders
que ha
of the
tenido este trabajo
mobilization that these
mucha
work
generación
has
de información
required,
puesta
a
en
lot
campo
of
en los
information
últimos años.
generation that has
Entonces,
been
la
placed on
relevancia de este
field during
tipo
this
de
past few years. So
resoluciones
the
que son de
relevance of this
carácter
type of
vinculante para
resolutions,
los
which
países,
have a
por
binding nature
supuesto que
for
nos da
countries,
un
of
empujón
course
más o
gives
un
us
brazo
a
más para
shove
poder
or a
caminar,
boost
para poder
forward
conseguir
in
apoyo
order to
técnico
walk
a nivel
forward to obtain
internacional,
technical
para
support at an
poder conseguir
international level, in
presupuesto
order to
también y
obtain
para poder
budget, and also
identificar
to
esta
identify
temática dentro de las
these
prioridades países.
thematic within
Siguiente,
the
por favor.
country's priorities.
Muy rápidamente, tal vez no lea, pero lo
Very quickly,
importante son los
perhaps you
colores.
can't read
Este es
this,
un
but the
resultado de
importance
un
is the color
análisis muy
code, and this is the
reciente que
result of a
hicimos
very
de
recent
cómo
analysis that we did
las
about
contribuciones de
how
la política
the
pública
contribution of
relacionada a
public policies
ecosistemas
related
ambientales
to mangrove
se
ecosystems
mezcla o se
gets
vincula con
mixed
las
up
metas
or
ambientales
links
en Costa
with
Rica y a nivel
environmental goals
internacional.
in Costa Rica and also at an
Las
international
líneas
level.
azules
The blue
básicamente es
lines
la
are
información
basically
o la política generada
information or
internacional dentro de
the
la
policies
Convención de
generated at an
Ramsar
international
y
level
vemos
at the
cómo
Ramsar
los grandes
Convention, and we can see
realmente
how the
aportan también al
arms also
cumplimiento de
support
otros compromisos,
the
como
compliance of other
las
commitments,
contribuciones
such
nacionalmente
as
determinadas, en
the
el caso de
nationally
Costa Rica,
determined
la política
contributions,
nacional
the
la
national
estrategia de
mangrove
conservación de
wetlands
manglares,
policy,
la
the
estrategia regional,
mangrove
tenemos una estrategia
conservation
regional
strategy, the
dirigida
regional strategy.
específicamente para el Golfo
We have
de
a
Nicoya,
regional strategy that is specifically
la
addressed for
Estrategia
the
Nacional de
Nicoya
Carbono
Gulf,
Azul,
the blue
lanzada el
carbon
año
strategy
pasado
that
en
was also
febrero, que ahorita
launched
les voy a
last
hablar
year
un
in
poquito más
February,
de
and I'm going to
esto.
mention a little bit more about it
La
in a few
Estrategia
moments.
Nacional de
The National
Biodiversidad,
Strategy
el
for
Plan Nacional de
Biodiversity, the
Adaptación y el
National
Plan Nacional
Adaptation
de
Plan, and
Descarbonización,
also the
¿verdad?
National
Esto tiene
Decarbonization Plan.
sentido a un nivel de
This makes sense at a planification level, of course, in order to be a little bit more efficient, which is something that we're still struggle with a little bit more at a state level. But we did this so that we could synergize in the operational end because it's still the same people working in the country. And secondly, in order to be more efficient in the transparency and execution of national budget and of international cooperation.
And as I was saying, we mentioned the contributions that are determined nationally. How does this work here in the big subject for restoration? How is this linked with the communities, for example, that are in Oncotal? There are some commitments that are specific that were communicated in 2020 in the scenes of the ecosystems for blue carbon, and this comes from the accord from the Paris Agreement. And then, so first of all, we can see how we need to understand how although this is a very technical issue for system restoration, but how that can also be linked to a public policy and an international policy as well. So thanks to this, Costa Rica improved its relationship with other governments, such as France. In the last years, we have executed many actions that support the the establishment for example, compliance for wetlands. And then we also have a baseline to continue monitoring and identifying priorities. The other great achievement that we have is the launch of the National Strategy for Blue Carbon. This was launched last year in the framework of the National Day for Wetlands in February. And this, more than a document, has shown the relationship between the documentation and the generation of public policy and international agreements, and how that is being reflected in fieldwork. From one document to be able to show results, as Elenin was demonstrating, that's not easy for every country. And Costa Rica, with a lot of efforts and also many challenges, but with the support, an increasing support in the last year, we've been able to establish that link, that alliance, and to determine how we can use the elaboration of a public policy to show how we can include communities in different areas. Communities have had a very important role in these last years. Without them, we wouldn't be able to actually be here because they help us to process data, organize and reorganize. There's a constant assessment, and this is the relevance that of what we've been sharing with the world. In Latin America, there's been a lot of interest to know the experience that Costa Rica has had, not only with not only in the establishment or making one more document, but how we have worked with politics and have an indicator for restoration. So a little bit to show you a little bit. This is important. The generation of material to campaign, to divulge our knowledge. This is a carbon blue knowledge that we know. Sure, we all of us might know this, but not everyone does. And with this slide, I would like to focus on two specific messages. Generate messaging and communication is completely relevant to advance. Clear information, basic concept, and to be able to streamline the information and have that reach the population. And then number two, you can see the red circle shows those places where Lenin showed us that are
de
in the
restauración
initiatives
de
for
moneda por
restoration
razón
for
en el
blue
país.
carbon
El más
in the
grande,
country. The
que también
largest
nos fue
one,
comentado, por dicha, yo voy
which
ahorrando ahí
was
minutos
also
porque ya
shown,
los ponentes
I'm
anteriores
kind of
me ayudaron. Pero el círculo
saving up on
volcán
time
corresponde al
because
humedal estero Punta Arenas,
they
donde hay
also
una
talked
tarea
about this before.
bastante
But
grande, no
the
está
largest one
finalizado.
is a wetland in Punta Arenas
Pero
that
ha sido
isn't
uno de los proyectos
finished, but
más,
it's been one of the largest
más retadores, por
that's
así
had most
decirlo, que hemos tenido en el
challenges
país. Pero bueno, la suma,
of what we've had in the
la suma
country,
de
and then the
todos
sum
ellos
of all of
nos ha
that
permitido, ¿verdad?,
has
de ir
shown us and
mejorando y que
allowed
los
us to
siguientes, los
improve,
futuros, en el
and
corto
that
plazo,
the
proyectos
the next
puedan
people that
tener
are in
muchísima
these
más
projects
claridad.
can have
Y
more
eso quiere decir
clarity,
también
and that also
reducción de
means
costos. Siguiente.
cost reduction.
Voy terminando.
I
No
will
voy a
be
entrar
finishing.
la,
I won't
en la
really go
descripción
into
de
the
esto, pero
description
solo
of
quería
this, but I
compartirles
just
que
wanted to
esto es
share that
parte
this is part
de la
of
guía que tenemos
the
en el
guide
equipo
that we
nacional.
have
Y cuando
in
hablo de
the
equipo
national
nacional,
team.
el
And when
SINAC,
we
la
say national
academia,
team,
son
that's
las
the SINAC,
ONGs, son las
the
comunidades,
academies,
es a
the
nivel político,
NGOs, the
es a nivel de
communities,
municipalidad, ¿verdad?
politics,
Realmente es un equipo
municipalities.
nacional que funciona así.
It's a
Y
national
cada quien
team that
tiene
works
su
like
rol.
this, and
Este
each
esquema
one
nos
has
ayuda a
their
no perder
own
el camino,
role,
porque
and
muchas
this helps
cosas
us
van
to
sucediendo.
not to
Por
keep
ahí
our
está
way and not
el
get lost,
anaranjado, los
because
mecanismos financieros de
in
mercados
orange
no
we have
mercados. Ese es un
the
tema que ahorita es el que
distribution of
necesitamos
benefits
desarrollar
and
un poco
funds,
más, ¿verdad?
non-market
No está
mechanisms and market
concluido, no está
mechanisms
definido en el
that
país.
isn't
Pero
concluded, that
este es el
isn't being
engranaje para
defined. But this
mostrarles
is
que realmente una sola
the
institución
way
no
it all
puede
works,
abarcar realmente un tema
and we
que
can
podría
see
decirse solo
how
la
these
restauración de
gears
ecosistemas. Así que a nivel nacional
link
ya se han
all the
identificado los
institutions,
roles
and international
y las
level we
rutas también
understand the
para
different
poder tener
roles
resultados
and different
eficaces.
pathways that we need to
Siguiente para
follow.
finalizar, creo.
And the next one to kind of
Sí, esta
finish,
es
I
mi
think.
última
Yes, this is my
diapositiva.
last slide.
Las imágenes dicen más
Images
de
say
lo que yo les
more
pueda
than I
contar.
can say, really.
Esto es la
From
primer foto
politics
es
to
el Humedal
action.
Nacional Terra
First,
Baciense.
first picture
Básicamente
is
es un sitio,
a
nosotros
wetland
les
from
decimos
Terraba
pacientes, todos los
Sierpe.
manglares afectados son
We
pacientes con condiciones
we say
diferentes
that they
y por
are
ende
patients,
necesitan una atención
wetlands that have
especializada,
been
individualizada y también tienen los
affected.
costos
We say
pueden
they're
variar,
patients,
¿verdad?
and
Y
they
las
each
necesidades y
need
las
their own
alternativas pueden
specific
variar.
care.
El
Costs may
humedal Terra
vary.
Sierpe es el
A lot of
tema de
things
una
may vary.
especie
And
más
this first
agresiva, ¿verdad? Que
wetland,
va a
there
ir
is an
colonizando, que es la
aggressive
negra forra.
species, which is the
Tenemos un
negra
proceso
forra.
bastante,
That's a
bastante
specific
avanzado. El segundo
species.
es el
And then
humedal
the
estero Punta
second
Arenas,
is
donde ya había
the
caña de
Punta
azúcar y ahora
Arenas estuary
tenemos esta
wetland.
regeneración
And we
natural
have
de
this natural
casi 5 o
regeneration
6 metros.
of almost
Y el
5 to
último
6
es Bahía
meters.
Thomas en Coajimalí,
And then the
donde por
last
más
one
de
is
25
Bahia
años fue una
Thomas
sandunguera
Bay in
abandonada donde absolutamente no había nada. Era,
Guayaquil, where there
funcionaba
wasn't
casi que como
anything
una
there.
cancha de fútbol y
It was
ahora
almost a football
tenemos un
field,
proceso
and now
bastante
we
avanzado
have
también.
an
Entonces,
advanced
es solo una ilustración
process. So
de
it's just
cómo
an
desde la
illustration
política
as
podemos
to
lograr
how
estas
we
cosas.
can
Son procesos que
reach these
no
things
se
from
terminan
politics.
en 4
These
años
are things
ni
that
en
don't
5
really
años, ni en un
end in
gobierno,
4 to 5
¿verdad?
years
Son
or in
definitivos y
one
bueno, este es el reto,
government
una
alone.
invitación
There is a
para
challenge,
los
and
que no se han
this is an
unido
invitation to
y
those that
desde la
haven't
academia
been
también decirles
involved
que hay
in this, and
muchísima oportunidad
from
para que
our
logremos investigación,
academy,
resultados.
to let you know that there's a
Muchas
lot of
gracias por la atención.
areas to get involved in, from research to studies, volunteer.
Gracias a Jackie.
Thank you.
Ahora abrimos
Okay,
el
thank you,
espacio.
Jacqueline. Now we
Tenemos
will open up the floor for some questions. Yes, thank you for the presentations. They have been very clear. My name is Alexander Kame. I have a project with monitoring of the red tide in Costa Rica using satellite imaging, and I thought this was very interesting as to the impact due to these micro seaweed and what you mentioned with the coral reefs. My question is, what is the potential do you see in monitoring for the environment using different technologies? Does anyone have experiences with this in this work, or have you used any type of service, data service to monitor that, um, any type of changes you've had in the ocean. It's a general question, but I guess the impact for micro seaweed and what you've mentioned. Okay, well, yes, there are some experiences, and in fact, right now we have a student with performing their master's, and he's studying the mangroves with drones and different sensors, remote sensors. And one of the challenges we have is that as we advance in the restoration, how do we monitor that? Because the field, that would be very expensive to monitor that in the field. And in the Punta Arenas project, we estimated that only the monitoring would be from 10 to 15,000 annually if we do that by foot. So then that's when we started to think about this thesis where it's almost finishing, where we wanted to establish different satellite technologies, remote technologies to try and find a balance between the species, the areas, and the sensors. So the student finished the gathering all of the data, and so we need to look into the restoration, different species, and we want to move in that direction to automate the monitoring of the restoration of these wetlands through sensors. Yes, and directly, the student that's doing this is actually just left, but what we're using is cameras. We're taking pictures and reconstruct that in 3D. So what our idea is, as time moves forward and with the restoration, maybe we can see the 3D version of the coral reefs. And maybe to amplify a little bit more in CNOC, sure, there are much facilities in trying to obtain imaging, satellite imaging by high resolution through international cooperation. We have access to that, and that has allowed us to include within the protocol for mangroves the using Google Earth Engine. There is a procedure to really take advantage of that monitoring and to kind of measure that, but we are moving within that direction. We want to be cost effective, lower costs, and because humanly, we're not enough, and we are in 2024, so we want to use technology. Yes, I would just like to maybe say to point out that sure, it's easy to see the mangroves that are exposed in those environments or the surface of the water. But when we talk about different ecosystems that are maybe in more turbid waters, more deeper waters, or some fields are very shallow, it's not as simple. There are many corrective techniques and a lot of things we can do, but I, for example, worked with Australian colleagues, and I can clearly see that I'm on a boat and I can't see. I can't just like move my hand and see that. Any other questions? Yes, thank you for the exposition. It's very clear, very interesting. I wanted to ask Jacqueline, from the perspective of public policy and different interests that we have in coastal borders, what are the key areas that we want to show for restoration when there's such a high pressure for development?
Esa es la pregunta más temida.
That's the question that we all fear, and that's a reality.
la realidad es
The
que
reality
los
is that
impactos
the
están
impacts
pasando más rápido
are happening
que lo
quicker
que la información de
than
monitoreo
the
que tenemos
information for
disponible.
monitoring that we
Entonces,
have
ese es el
available.
primer reto,
So that's the first
realmente
challenge,
mejorar y vuelvo
to
a la pregunta
improve.
anterior con el
And
tema de
I
la
go back
tecnología,
to the
¿verdad?
previous question with
Fortalecernos un
technology.
poco
We need
más,
to
tenemos que ser más
fortify
rápidos,
that
reto
area and be quicker,
institucional que no lo
and
tenemos
that's
que resolver
a
solos, eso es
challenge
importante,
that
¿verdad?
we don't have to resolve
Por eso es que
on our
tenemos
own, and
tesistas
that's
en
important. And
Edcate, por
that's why
eso es
we have
que
people
Amanecia ha
working
ido
in
ahí,
different
¿verdad?
theses, and
Coordinando muchísimas instancias,
there has
pero
been
también otro tema
a
es
hand
la
within.
generación de
We have different
política en
help
otras
in different
ramas, por
instances,
ejemplo,
but
en
also we see
agricultura. Hay
policy
política pública
in different areas, for
que se
example,
genera,
agriculture.
¿verdad? Para apoyar la
There are
agricultura y la
some
ganadería y la pesquería
policies that are
que
set
no
up to help
necesariamente
cattle
coinciden con la
and
política
fishing
pública
that
ambiental.
might not
Ese
coincide
es un
with
reto que no
public
solo tiene Costa Rica,
environmental
tiene
policy, and that's
muchísimos
a
países y es
challenge that not
cómo
only Costa Rica has,
enfrentar,
many countries
cómo
have that. And how can
igualar,
we face,
¿verdad?
how
O
can
cómo
we
sincronizar que la
sync
política pública
those
se
two
genere de
policies,
una
public
forma
policy,
integral,
and
no
then
por agendas específicas,
not
porque
only
al
by
final todos vivimos en el
agendas,
mismo
but in an
territorio y los
integral way, because
ecosistemas
we
están
all
en el
live in
mismo
the
territorio.
same
Entonces,
area, same
esa es
territory. The
la
ecosystems are
respuesta a
right
eso,
here,
yo
and
todavía no la
the
tengo
answer to
después
that, I still
de
don't have that
una década de estar
after a
trabajando en
decade
esto.
of
Se hace
working
lo
with
posible
this.
para que los espacios de
So
generación de
we see
política
how it's
pública
important
se pueda,
in the spaces
¿verdad?,
of
hacer
public
en marcha.
policy to kind
Pero
of
sigue
match that
siendo
up, but it's
un
still
reto
a
bastante
challenge,
difícil. Aquí el
a
tema
tough
es
challenge.
acelerar y
And
aprovechar
they
la
are
tecnología
accelerating,
disponible
and we
para
want
poder
to
captar
use the available
aquellos
technology
impactos
to
o
capture
aquellas
those impacts
presiones
and those
para que
pressures
no lleguen a ser impactos.
so that they don't
¿Verdad?
become an impact.
Vamos a ir trabajando día a
So
día
yes,
con
we're
esa
working
tarea.
with that day to day, with that challenge.
Nada más para
And
pequeño comentario
to
al respecto, yo creo que el modelo
a
de
small
desarrollo
comment
también
on
hay que
this,
repensar,
the
especialmente
development
para
model,
los
we also
zonas
need to
costeras.
rethink this, especially for
Entonces, apuntarnos a un
coast.
desarrollo integrado,
We need to bet more for an integral development
toma muchos elementos
with integral principles that kind of captures many elements, not just here.
de otras discusiones,
We've had other discussions, and we've seen this
los problemas,
because the issues,
ambientales,
the environmental issues in our
costeras,
marine and coastal areas have become quite complex. So to approach that, disciplinary approach isn't enough.
pasa por ahí.
So we kind of
Si hay alguna
need
otra
to look into
pregunta,
that. Is there any other question?
bueno,
Okay then.
yo les agradezco,
I want to thank everyone
su presencia,
for your
participación.
presence,
Un
your
aplauso para los
participation.
panelistas.
A round of applause for the panelists. And
Tal vez nada más como reflexión
just as a reflection,
que le toca normalmente al
which
moderador,
usually
apuntalar
the
algunas ideas.
moderator kind of
Yo creo que
says
es un tema,
this, but
es un
this is
tema
something
nos
that
recuerda
reminds us
que hay que recuperar
that we need to recover
a través de estos procesos
through these different
lo que ya se ha
processes,
ido perdiendo.
different
No
procedures,
logramos
what we've
conservar.
lost and what we haven't
Entonces,
been
como que
able
vamos cambiando las
to
palabras,
to
¿verdad? Ya no-
conserve. So we need to
ya,
change our
tenemos
words.
que pensar en nuevos procesos
We need to
que
think
nos
of
permitan
a new
través
process
de-
that we can,
la participación
through
ciudadana,
the
a través de la política
participation
pública,
of community
a través del
public
conocimiento
policy,
científico,
through scientific knowledge,
a través de tomar en cuenta también
and after
elementos
taking
externos
different
que,
external
como el
elements
cambio climático, que
like climate
también
change,
hace su aporte,
that
que nos
also
obliga a
gives us
repensar muchas cosas,
a lot of information that makes us rethink a lot of things. And this is regarding an experience
Entonces, es
um,
interesante
as a country. So it's important.
Lo que he visto es hay muchas iniciativas de restauración.
We've seen a lot of initiatives for restoration, and I think it's important Yes, we need to recover.
volver
We
darle
need to
funcionalidad a los ecosistemas, hay que
give
tratar de
functionality
ayudarlos,
back to
tratar
these
de
ecosystems, help
su
them,
capacidad resiliente,
even
pero
if they are very resilient.
ese aspecto me llama la atención
I
y
feel it's always
yo creo
something
que
that
hay mucho
I
espacio en el futuro
think
para
it's very
seguir
important, and there's a lot of area of
impulsando
growth
a través
in the future that we
de la
can
política
impulse through
que
policy,
desarrolla el Estado.
politics,
En ese sentido,
from
pues,
state.
coincido mucho con Jacqueline, esto es un
And I
trabajo
share
entre
that with
todos los
Jacqueline. This
actores,
is something
¿verdad?
that all of the
Tenemos
actors
que
need to
aportar
get
desde
together
nuestras
and
diferentes especialidades o
provide our
nuestras
areas
diferentes
of expertise
trincheras, pero de manera muy concertada.
in a very assertive
A
way.
veces hay otros elementos
Because there are other elements
que son importantes
that are also
en
important
todo esto también
in
es
all of this,
fundamental
and all of this is
tener
fundamental.
claro cómo
And we need to
vamos
be clear
nosotros
as to
a garantizar
how we are going to
a las
guarantee
futuras generaciones realmente
future
la
generations are going
que
to
tuvimos nosotros.
understand what we've had and what we
Así que
will have
los
in the
invito
future. All right, so I will invite you
almuerzo,
all to
que
lunch.
tengan
Have a
muy
great
buena tarde.
afternoon. Yo creo que ya. ¿Me harías ayudándome a...?
¿A desconectarte?
Ajá.
Sí, ¿cuál es el tuyo? ¿A dónde
Ese.
estás?
Lo pusiste.
Ah,
Ese
ok,
le no.
ok.
Ajá. Excelente. ¿Y sacaste el día entonces? ¿Ya lo tienes libre?
Sí,
Ay, sí, está bueno.
sí, ahí vi también el mensaje. A mí también me dijeron que el fiel era el de un día entero. Pura
Ah,
vez.
¿te dijeron?
Sí, yo